are the still unknown plant growth regulators. That recently the government has brought in regulation to market the biostimulant in the agriculture market, and this is one of the emerging areas where many startups are coming in. Already existing industries also pushing to take advantage of the biostimulants. So the plant growth regulators, the biostimulants, I believe they're different from the plant growth regulators, because in plant growth regulators, we know what is the actual molecule that is regulating the plant growth regulators, whereas in biostimulants, it may be a single substance or multiple molecule present in that biostimulant formulation that may regulate the plant growth and development and ultimately improve the yield. So there is a lot of research that can be done in plant growth regulator for their application in improving crop productivity and entrepreneurship development, and also from the biostimulants, which are several categories that are at the end of my presentation. I will just touch upon them. And there also a lot of work can be done to discover the biostimulant molecule from each of the category, and then further fine tune it to improve the crop productivity. So this one I will start with the abscisic acid. As I told, they have been associated with this abscisic acid work for some time. So this is as a case study, I project this. This can be hold good for any other elicitor molecule or any other the signaling pathway. So this abscisic acid, it was discovered as Darwin or abscess in 1960s. And from either the upsizing bolls of cotton or from the buds of the sycamore buds which undergoes dormancy, this compound was initially isolated, and it was named in 1960s as abscisic acid. The interest for the plant scientist came in this hormone. Lot of interest was generated because of these papers. They showed that the abscisic acid can inhibit the transpiration, and if from the detached leaf, if you keep detach the leaf and keep it in the laboratory condition, the abscisic acid concentration increases drastically. These triggered a lot of interest, because in agriculture one of the major input is water, and as we understand, most of the water the plant takes up is used in the transpiration. So if you want to improve the water use and improve the water use efficiency and reduce the water use for unnecessary transpiration, even when the endogenous carbon dioxide content is sufficient for photosynthesis, this mechanism should be very tightly linked to the carbon assimilation and internal carbon dioxide concentration. So this attracted a lot of interest, and several scientists across the world, they were interested in identifying either how the abscisic acid trigger stomatal closure and how it induces the stress tolerance, and then how we can use it for improving the stress tolerance of the plant and other activity that ABA mediates. This is a textbook information. You can find in your textbook. This is the most biologically active abscisic acid, and other isoforms. They are partially active in some assays, or they are inactive. So abscisic acid, it plays diverse role. Mainly it imposes regulate transpiration, that is stomatal closure is mainly mediated by abscisic acid, and it is also required for vegetative growth. Some minimum concentration is required, otherwise the plants will not grow properly. This is a ABA deficient mutant. You can see that this mutant will not produce as much biomass as the wild types. So it is required for the normal growth. It is required for seed development, that is to acquire and assimilation of the storage reserves, and acquirement of desiccation tolerance, acquirement of dormancy. So all the activity is mediated by abscisic acid. And abscisic acid at low concentration, it promotes root growth. So it is also required for deep rooting, for water mining under water deficit stress condition. And again, it is necessary for the desiccation tolerance. That is, when the stress is extreme, the plant can survive if it has better abscisic acid signaling. And it also plays very important role in biotic stress signaling, as we can see from the rice brown spot disease. If with the presence of the abscisic acid, the plant get resistant to rice brown spot. So in general, it is believed that for some of the disease, at least the ABA plays very important role in providing protection. Besides this, it is also play a very important role in fruit ripening, synthesis of secondary metabolites. So it plays very diverse role. So if we can develop a novel molecule which can act like abscisic acid, or we can design a receptor which can be activated by off-shelf agrochemical, it will be very useful for the industry. And so for this purpose, the elucidation of signaling pathway was very important. So this was not very easy task. From 1970s, it was known that this abscisic acid plays very important role in stress and other activities. So several labs in the world, they started doing research to understand how the abscisic acid triggers all these activities. And they were looking for a receptor, as we look for other hormones. For this also, they were looking for the receptor. But till 2005, no receptor could be identified, although several components have been identified that they play a very important role in stress tolerance. So in 2006 to 2008, at least 3 independent publications appeared on the discovery of abscisic acid. From Robert Hill's lab from Canada, he reported that the RNA binding protein FCA is an abscisic acid receptor. And then from CIU, the magnesium lattice H subunit, which is located in the chloroplast, was reported an abscisic acid receptor. And another group reported that a G protein coupled receptor act as a abscisic acid receptor. But within this short period of time, several other labs, they started working on these receptor, and they found that none of these receptors could be like proved as a bona fide receptor of abscisic acid. So this was like in 2008, Peter McCart and Krillman, they wrote a like opinion paper in Plant Biology. The title says the status of ABA receptor discovery at that time. So then, so the search continued. In 2009, from Sarah Asman, she reported that the G protein coupled receptor type G proteins, 2 G proteins she identified, and they reported this as the receptor. And they could show that if there is a double mutation in the 2 proteins that are there in the Arabidopsis, they are relatively insensitive to ABA-mediated inhibition of germination. That is, if ABA is used in the germination medium, the germination is inhibited with a simple bioassay for abscisic acid. If you have a double mutant, they are less sensitive as compared to the wild type plant, like they are sensitive to the abscisic acid. And this phenotype was the only phenotype that they observed, and also they could find a mild phenotype of stomatal aperture closure. That is, if you apply abscisic acid, the stomatal closure is there in the wild type, whereas in the double mutant, the closure is relatively less. So based on this, they proposed that this abscisic acid, the GTGs can act as a ABA receptor, and they regulate stomatal closure. But still, a lot of evidences are avoided. So after this study, that time the major limitation was like only about 1% of the GTCs present in the assay medium, they could bind to the abscisic acid. Like it is not the one-to-one binding was not there. And in the animal equivalent of this GTCs, there were ion channels. So there were still a lot of questions whether it is really acting as abscisic acid receptor or not. So then in the same time, these two groups were independently working on discovery of abscisic acid receptor, although both of them, they used different approaches. Erwin Grill's lab, they used uh, uh, this protein phosphatase 2C, which are well-known negative regulator of abscisic acid signaling. They used the ABI as one of the bait protein to do yeast-2 hybrid assay to find out the interacting partner. So if we find out some interacting partner, they expect that the ABI may be interacting with the receptor. So we can identify the receptor, and they were successful in identifying the protein that can regulate the protein phosphatase 2C's activity. And another group from the University of California, Riverside, Sam Cutler's lab, they were looking for the ABA receptor by using a chemical screening approach. So earlier, their concept was abscisic acid is a pan-agonist for all the ABA receptor, like whatever will be present in the plant. So if we use some chemical which may be act as selective agonist for one particular receptor, and if it is not inhibiting all the receptor, then only we'll be able to find out a mutant which is lacking any ABA response. So they used this chemical screening approach. So both of the lab, they found out the same proteins. So by using yeast-2 hybrid assay from the Grill's lab, they identified the regulatory component of ABA receptor. They named this protein as RCIR. These proteins belongs to the family of start domain containing protein. These are small protein having amino acid residues of about 159 to 220 amino acid. In Arabidopsis, about 14 family members were there. So this explains that why a conventional mutant screen is not able to identify a receptor mutant, because if one of the receptor is mutated, the other receptors can compensate for the function when we use abscisic acid as an agonist. So they could identify this receptor, and they have shown that in the germination assay as well as in the stomatal closure bioassay, if we mutate these receptors, or if you overexpress these receptors, in the assay, it responds to abscisic acid. This one was from the Sean Cutler's lab. They used a chemical called pyrobactin, which specifically inhibit the germination, and it mimics the ABA response during the germination process, not in the other activities. So they used this pyrobactin, and they identified a mutant which they named it as pyrobactin resistance 1. And when they, by use map-based approach, when they cloned, it was same start domain protein that was identified by the Erwin Grill's lab. And by using in vitro assay, they showed that the pyrobactin 1 interact with the protein phosphatase 2C, that is hypersensitive to ABA1, or it is ABA insensitive 1 or ABA insensitive 2, in the presence of abscisic acid. So the abscisic acid is necessary for interaction between the receptor protein and the protein phosphatase 2C. So this was identified because earlier it was known that the protein phosphatase 2C, they negatively regulate SNRK2 kinases, although the detailed mechanism was not known. It was known that this protein phosphatase 2Cs, they regulate SNRK2 kinases, and SNRK2 kinases, they in turn phosphorylate the ABA responsive element binding transcription factor and other proteins that are involved in the ABA signaling. So based on this, these two groups, these papers were published back to back in the Science in 2009. And this was the reaction from the different scientific group, and this paper was published. So most of the biologists accepted that these are the best evidences, these two papers, and still some more evidences are needed to finally prove that these are the bona fide ABA receptor. So again, several groups, once these papers were published, they were working on to reconstitute the ABA signaling pathway. So once if these are the receptor, if you reconstitute ABA signaling pathway, then it will come as a proof. So Jiang Kangsu, who was also associated with the San Cutler in the earlier publication, their labs are almost, it is in the one lab, one side Jiang Kangsu's lab, the other end was San Cutler's lab. So they were trying to identify or trying to reconstitute the ABA signaling pathway to identify all its core components. That is, if we give the elicitor, how the gene expression or any other response is mediated, whether we can identify all the components and reconstitute the signaling pathway. That was the major question. And there are 14 members are there. Unusually, when compared to other plant hormones, which have like 2 to 3 to 4 members as a receptor, in this 14 members were there. So we wanted to know like all the members, whether they act similarly in the ABA signaling, and what is the structural basis, how ABA binds or how these component interact, and is there a natural antagonist for this receptors. And the structural part was work was carried out by Van Andel Research Institute by the Eric Shu, and the in vitro reconstitution work was carried out by Jiang Kangsu lab. So that time I joined his lab, and Dr. Zhu suggested me that if I can do the carry out work on the reconstitution of this abscisic acid signaling pathway. So I selected this Arabidopsis protoplast system because this is the easiest one to experiment several proteins to see if they interact and if they transduce signal from one to other. Because we can, if we have a good reporter system, we can demonstrate that by addition of one or more protein, how this reporter system is activated. So here I used the ABA activated gene, that is RD29B promoter, as a reporter gene, and it was fused with lug gene. And in the absence of any of the ABA signaling component, we don't expect the expression. And if we add any of the component that we suspect that it may work in the ABA signaling pathway, we expect that the signal may be on. So this is very easy system to decipher the function of multiple protein. So I am thankful to Professor Jenshins. He is a pioneer in the protoplast transient expression system. She taught the entire system how to use it for deciphering the signaling pathway. So by using this system, we tried several components that were known by that time. And we used a protoplast system which is prepared from the SNRK triple mutant, because that is one of the positive regulator. If we have a SNRK triple mutant, that is less background noise in the ABA signaling. So we used that protoplast to try different components and see whether our reporter is activated or not. So this is the ABF2 transcription factor, which is known to activate RD29B promoter. So in this protoplast background, if you add ABF2 and if you add abscisic acid, if it is a normal protoplast, we expect that the ABF2 expression will be induced. But since we used the SNRK triple mutant protoplast, there was no induction. So it was a perfect system to study multiple components. And then we add SNRK 2.6, which is a kinase that phosphorylates ABF2. We can see the induction of the expression of the stress responsive gene. And if we add the protein phosphatase, any of the protein phosphatase of the 9 protein phosphatase 2Cs that are present, the induction is repressed. So somehow the ABI1 could repress the activity of SNRK 2.6. And then we added the ABA receptors that are identified in the Arabidopsis. There are 14 genes are there. One by one, we tested whether which can reactivate the expression. And we found that the addition of PIR1, it activated the expression of the reporter gene, that is the signaling is on. And if you use the mutant version of the PIR1 receptor, then it is not working. This was original mutant that was identified from the pyrobacterium resistant 1 mutant screen. Or if we use the ABA1 dominant mutant, then also the activation was not there. So with this, we could show that if with the after testing multiple component, we could show that this ABA signaling pathway has only 4 core component. One is the receptor, and then protein phosphatase 2C, and then SNRK2 kinase, and finally it is a transcription factor or any enzyme that is activated by SNRK2. So in the presence of ABA, in the absence of ABA, the protein phosphatase 2C repress the activity of the positive regulator, whereas if we add the ABA receptor and if we add the abscisic acid, it relieves the repression of the abscisic acid insensitive 1 and other protein kinases on the signaling pathway. So with these 4 core components, we could show that the pathway can be reconstituted in the Arabidopsis protoplast. And I have tested all the components, various the receptor that are present. Here you can see the pyrobactin 1 to pile 12. There is one more receptor, pile 13, and that story I will tell later because that was not active in this assay system. And we showed that all the 13 receptors, they could work in the same way. And we tested with different protein phosphatase 2Cs also, and we could show that this system worked with different protein phosphatase 2C. In the in vitro condition, it was shown that if we add in an abscisic acid dependent manner, the ABF2 phosphorylation can be demonstrated in the in vitro condition. That is, you use recombinant protein, and only in the F1W you can show that the ABF2 phosphorylation can be in a concentration dependent activated by the ABA receptor and abscisic acid by overcoming the effect of the ABA1. So this proved that the abscisic acid receptors that were identified were real bona fide receptors, because we could show it in the single cell system as well as in the in vitro system that it could activate the gene expression, or it can mediate the phosphorylation of this ABF2. So this showed that these are the 4 component, and this the pathway can be reconstituted, and this similar system or this system can be later it was later used for various other purpose to discover other novel molecule. In the other study where the crystallography study was carried out to know how exactly the abscisic acid and the abscisic acid receptor and the protein phosphatase, they interact. So in this study, the crystal structure was deciphered, and the important residues that interact with the abscisic acid was identified, and the important residues that interact with the ABA receptors were identified. And again, by mutating those specific residues in the protoplast assay, it was demonstrated that these residues are very important for the binding and activity of ABA receptor. So all these things led to publication of these studies in Nature 2009 December issue. In this issue, 4 articles were published. And based on this, this signaling mechanism was proposed in the same article. It is in the absence of abscisic acid, the protein phosphatase 2C, they repress the SNRK2 kinase mediated activation of a downstream component. In the presence of abscisic acid, the protein phosphatase 2C is sequestered by the abscisic acid receptor, now the SNRK2 kinase is active and it phosphorylate the transcription factor and other protein to activate the downstream signaling. So this that time it was like rated as a top science breakthrough. And we continued that work further. And as I told, one of the receptor that PIL13, it was not activated by the abscisic acid, or it could not relieve the repression of the protein phosphatase 2C. And when the structure was compared, some 3 of the residues in the important critical domains, they were altered. Because of this reason, they could not bind to protein phosphatase 2C and relieve the activity. And if you mutate those critical residues, either double mutant is sufficient to regain the activity of protein phosphatase 2C. So this suggested that the receptors can be engineered to modify its activity. So knowing so much, at that time, lot of interest again on how to improve this discovery, use this discovery to improve the drought tolerance and water use efficiency. So there were 3 different approaches were taken by different groups depending upon their capability. One is engineer the ABA receptors or its core signaling component to improve the stress tolerance. This is a transgenic approach, simple, straightforward approach. The other one is alter the receptor. That is, you know exactly how ABA receptor signaling pathway works and how ABA interact with the ABA receptors. So you can engineer the receptors that can be activated by off-shelf chemical that are already available. That is, instead of abscisic acid spraying in the field, you can use any of the off-shelf chemical that is already available in the lab that can be used to activate the receptor. Or you can also identify a cheap agonist, that is ABA is a pan agonist, and ABA, as I told in the earlier slide, it regulates diverse activities. So if you use ABA as a spray, several activities are induced which may or may not be necessary for that particular condition. And if we want to improve only stomatal closure, then we have to activate only certain ABA receptors, that signaling pathway to activate the ABA receptor. So for this, also you can use some of the cheap agonists which can be discovered by using this kind of single cell assay system. So we have engineered rice with this ABA receptors. We cloned 10 ABA receptor from rice, and we have engineered, and we found that different ABA receptors can give tolerance to different kind of stress. That is, all the ABA receptors are not working in the same way. So today I'm not talking about this component. Only some example I want to give from these 2, because these are related to the plant growth regulator based entrepreneurship development. So this was one of the dream from long back, that is whether we can vaccinate the plant against stresses. And yes, if we can use abscisic acid, it can be vaccinated and the plant become tolerant to stresses. But because abscisic acid is a pan agonist for all the 14 receptors that are present, so if we activate all the 14 receptors which are involved in the diverse function, then it may affect the plant growth, even if it improves the stress tolerance. And also, abscisic acid is costly chemical which may not be useful as a like a spray in the field, or as in this case they used along with the irrigation water. So several groups, they were trying to identify ABA agonist or ABA receptor agonist and antagonist, and engineered ABA receptor that can be activated by off-the-shelf agrochemical. So when I was doing protoplast assay, I found that the pyrobactin 1, which San Cutler's group identified as agonist of the abscisic acid receptor, it was not working as agonist for the different kind of the abscisic acid receptor, different family members that are present in the Arabidopsis. So in this case, if you see either in the addition of the pyrobactin or addition of abscisic acid, the ABA-induced gene expression is enhanced when the pyrobactin resistant 1 was used as a receptor. But if you see in this one, the PIL2, when it was used as a receptor in the assay system, in the addition of abscisic acid, the activity was high in this one, but as the concentration of pyrobactin increases, even the abscisic acid induced the gene activation was reduced by the pyrobactin. And only if you use pyrobactin, there is no induction. Here you can see if you increase the pyrobactin concentration, the stress response to gene expression increases. So this demonstrated that the pyrobactin 1 is act as a agonist for some receptor, and it has act antagonist for other receptor. And when the structural studies were carried out, we found that some critical residues are there that alters the binding between the pyrobactin and the receptor. And by using in vitro mutagenesis, we have shown that conversion of a single amino acid residue can convert the PIL1 from the pyrobactin activated to pyrobactin inhibited receptor, or the PIL2 can be converted from the pyrobactin inhibited receptor to pyrobactin activated receptor. So several of the pyrobactin analogs were synthesized, and some of them they found very effective in activating the ABA signaling pathway. So this then again these kind of studies continued. Some other group they found came out with other molecule which is they named as quinobactin, and this quinobactin, when it is treated to the different crops, either it is in the soybean or it is in the barley plant, they enhance the stress tolerance similar the way which is similar to the activation by the abscisic acid. So this kind of work it is in continuation. People are working to identify some of the newer molecule. The other approach was to identify the off-the-shelf chemical, whether any can be used to activate the ABA receptor. So in this case, what approach was used is first the existing ABA receptor was mutated to bind to the off-the-shelf agrochemical. Here in this case, mandipropamide was selected, which is a fungicide, and 6 amino acids were mutated in the ABA receptor. So this mutant version of the ABA receptor, which is called as PYR1 mandi, when it is overexpressed in the plant, this can now bind to the mandipropamide, and this will not be able to bind to the abscisic acid. So by using this transgenic plant, they have shown that if you spray the mandipropamide and the stomatal closure is induced, that is, this is the PYR1 like mandi transgenic plant, and when you spray the mandipropamide, it activates stomatal closure, and it also provides stress tolerance. That is what ABA is doing, that can be done by this orthogonal receptor, which is binding to the mandipropamide. So this kind of system is very useful if you want to regulate only a specific process at a specific time. You can use this kind of modification. That is, you use totally a ligand which is non-native and which is not able to bind to the normal receptor, whereas it binds to only the orthogonal receptor. So sometimes if you want to inhibit certain process that are mediated by abscisic acid, because ABA is also negative regulator of some of the growth and developmental activities. So in this case, they synthesized a molecule that can lock into the ABA receptor. But once it locks, there is a tail region is there that comes out from the gate and latch system of the ABA receptor. So now this cannot bind to the protein phosphatase 2C. So in the presence of this chemical, then ABA receptor will not be active, even if it is the normal plant. So this can also be used, like in case of strawberries, the ABA induces ripening. If you want to delay the strawberries, this kind of novel molecule can be used to delay the ripening. And this also induces, like abscisic acid pathway can induces some of the metabolite like antimycin. And if you use some novel molecule, you can increase the expression of this kind of novel molecule. So there are similar approaches can be used to discover the signaling pathway, that a single cell system, protoplast system can be used to discover like how the biostimulants that we are using in large extent, how it activates any specific process, or in the biostimulant, which molecule is involved in the activation of the specific process. That can be identified by using single cell system, and also to modify the proteins that are involved in the signaling, and to design novel molecule. This can be used. So these kind of activities can be like either biostimulant or use of the plant growth regulator based entrepreneurship can be taken up by the young students and other entrepreneurs. Now government is supporting this entrepreneurship activity. In our institute, we have the agribusiness incubation center. We incubate the startups, select the startup and incubate them, and also we will select them for funding. So almost last year about 36 startups were given funding, either the 5 lakh startup grant or the 25 lakh grant. About total 5.63 crores funding were given from the RKV Raptar project at IRI. This, in case of abscisic acid, there are a lot of future questions that can be addressed. And another important point that I want to say before concluding this lecture is that now government has come up with a regulation for biostimulant. Earlier, before the 2021, the biostimulants were marketed freely. And from 2017, several state government, they reported spurious product in the market, and also the biostimulant containing pesticides, lead, and other chemical. Hence, the Ministry of Agriculture and Farmers Welfare asked ICR to bring out regulation. So this regulation was brought out, and in 2021 February, where these 8 different classes of biostimulants are approved for the agriculture use. And recently, for 4 of these classes, even toxicity data requirement is waived off. For others, all kind of toxicity test is required, and for this 4 class, the toxicity test is not required. So the cost of development of biostimulant will not be very high. So I believe that many startups can come up based on the biostimulants in future. So the PGRs and biostimulants, they have a lot of potential to improve the yield and quality, as well as stress tolerance. And novel molecules, either natural or synthetic, agonist or antagonists of receptors, can be developed that can be used in the agriculture for improving the yield and stress tolerance. And again, design of orthogonal receptor, which can be taken up maybe large industries to come up with novel proteins, novel receptors for altering specific pathway. So with this, I would like to conclude my lecture by quoting this quote. It is the question that gets you the Nobel Prize. After that, you just add lab coat, coffee, and air conditioning. So this is very important. We have to really design our experiment. Then whether it is a student or it is a startup, our question is very important. If you know our question properly, then I'm sure the results will be there. So I thank the Department of Crop Physiology, US Bangalore, for inviting me to deliver this lecture, and also all the funding agencies. And one more quote: these two Nobel Prizes are waiting for any startups or students who is going to solve the problem of water. So one who solves the problem of water is worth two Nobel Prizes, one for science and one for peace. And saving water was also one of the dream of Professor M.K. Uday Kumar. So I believe the scientific community is going to work towards this, and it is not only like necessary for survival of the India. It is also survival for the world, because fresh water is a major dwindling resource worldwide. With this, I thank you everyone for your patience listening. If you have any questions, if you have time, I can take up some of the questions. Thank you very much.
Thanks for that wonderful lecture. See, we have read that kyolinite acts as anti-transparent. So how does it act? Is it through ABA or ABA receptor or some other different pathway?
So kyolinite is only through two mechanisms it is possible. One is it is acting as a physical barrier, and another one is it is reflecting the light that falls on the leaf. That is, it reduce the radiation load. So only these two mechanisms are at present known, and I am not like aware that this act through any other physiological process so far.
Okay, it's not related to ABA at all.
Yeah, it is not through ABA. It is not through any physiological means. So this is one of the debate again. Like we have in the biostimulant, one category is this, and there as anti-transparent. So in that again, some clarification we have given as per the definition. Whatever the biostimulant that we use, it should regulate some of the physiological process. If it is only through physical mode, that may not be come under the biostimulant.
Thanks.
One more question related to that, sir. I'm here. So much work has been done on abscisic acid, and then many analogs of ABA have been identified. But unfortunately, go to the field, none of those are have been extensively used. What would be the reason except the cost? Like sir said, when you say anti-transparent, you talk about only potassium chloride or kyolinite. Basically, that is a film forming type of anti-transparent. So ABA and its analogs have never been extensively used in the field.
See, use of ABA, it is not possible, because as I told, ABA regulates several things. If you use ABA concentration, even if it is slightly excess, then it is going to it may save water, but it will induce other physiological process that is going to reduce the yield, reduce the biomass production. That is why direct use of abscisic acid is not advised in any of the cases, although recently some application has come on the biostimulant that some industry has developed based on the abscisic acid, and they have shown that it improve the yield. But we have to target the novel molecule that either it is cheaper and it is not pan agonist for all the ABA receptor. It should be activating only some of the receptor, or the best one is use orthogonal receptor and use the other chemical, so that only the process that you require can be activated only at that point of time. It is not a general activation of all the ABA receptor.
Thank you.
So thank you, sir. It was a very informative talk. So this is Aisha, PhD scholar from crop physiology. So I was just thinking about fungicide that you are referring to. So I think it is fusicoxin, which promotes stomata opening. So in nature, if we have a disease symptom by any fungi or pathogen producing this fusicoxin, and you also spray ABA, so what would be the plant response then, sir?
So that fungicide that I was telling, it is not fusicoxin, but what you are saying is if the mode of entry is through stomato, then it may inhibit to certain extent, but I am not sure that that will be able to help in controlling the plant disease per se through stomatal closure, because that itself is not sufficient. There are several ways through which the pathogen can enter into the plant cell. So only stomatal closure may not be able to help. You can see in some cases, like when drought and disease interaction was studied, when drought is there for certain diseases, they found that this shows better resistance. The plant shows better resistance, where the mode of entry of the bacterial spore or fungal hyphae is mainly through stomata.
Yeah. So this fusicoxin producing
Pardon?
pathogen mainly enters through the stomata. So if we spray ABA, then I mean just a trade-off mechanism.
No,
like
I
that.
think if main mode of entry is through stomata, definitely ABA will help. But again, as I told, we need to decide all other concentration, like it should not induce the other process, so that the plant growth is not retarded. So plant growth retardation, if in our case, like whenever we overexpress abscisic acid receptors, we found that many of the receptor, when it is overexpressed, it reduces the plant growth, although it can increase the stress tolerance. So because of its multiple role, we have to target only specific pathway. If we can specifically close stomata by using maybe any other chemical, then it may be helpful.
Thank you, Vishwa. That is excellent. This is exactly what we were looking for. The genesis of this entire topic actually started when Professor Uday Kumar was here, and then we were looking at some of those chemicals which would perhaps act as agonists or antagonists for stomatal mechanism. When Ramu was here and his student was working on this, what we did at that time was to look for the whole process of the stomatal opening mechanism or the closing mechanisms, and then identify some of those compounds which, using the genomics approaches, find out a chemical that can now there and act. And then we actually started discussing with Praveen Vemula also. If there are some of these compounds which can be discovered for, like pyrobactin, for example, in ABA kind of mediations, is it also there for other hormones like auxins and cytokinins, where we can look for these kind of compounds, use the genomics approaches, find out a chemical which can go there and activate the whole process, and that will perhaps become much cheaper, and that will give a very nice opportunity for entrepreneurship development, was the thought. What's your opinion on that?
So I think chemical library screening will be one of the important approach immediately to identify certain antagonist or agonist. I am sure it is there. So maybe we have not put that much effort, but somebody puts an effort. It is possible to identify novel chemicals.
Yeah, that's the whole objective of this, and Praveen, we will discuss that later in the afternoon as well.
Sure.
Good afternoon, sir. As you said that you tested on Arabidopsis only. So myself, Pratik Sagar, I'm third year BSc Agri student here in GKVK. Sir, my question is that you have tested it only on Arabidopsis. What will be the response for other plants?
Which one you are talking?
Like ABA responses.
He says ABA responses was tested only on Arabidopsis. If you take rice or
the ABA signaling mechanism is highly conserved across the plant, and ABA is conserved even plants and animal. Even we also produce abscisic acid, although the receptors are different. the mechanism of AV action is same in all the crop plants. So if it works in one, in others also it will work, as it was shown in that soybean and barley, tomato. So already these chemicals, whatever works in Arabidopsis, it works in other crops also, because the pathway is conserved. This almost the ABA receptors, kinase, phosphatase, and the transcription factor, and this stomatal closure mechanism, all the genes involved, most of them, they are conserved across the plant kingdom.
One more question.
Sir, as you said that, myself Panbalagi from third year BSc Agriculture. So as you said that our main concern will be not like about rice, because food production and food scarcity is happening in our day to day. So if Arabidopsis is mainly interpret for barley and tomato, why can't we do that for rice also? Because it will be more helpful in increasing productivity in that one also, and then rice, wheat, and those things.
Yeah, certainly it will be working, because I worked only in this one. Definitely it will work in rice. We have worked with the engineering abscisic acid receptors. It gives increased stress tolerant to different kind of stresses. Definitely rice is the one which we need to work if you are interested in saving water. I agree with you.
Thank you, sir.
Yeah.
Hello, sir.
Yeah.
I'm Deepika from third year PhD from Department of Plant Biotechnology, sir. So I was just wondering, can we use these biostimulants for tissue culture? So are there any reports, like, certain biostimulants which are already reported, which can be used in tissue culture process?
So I don't know, maybe if any other can answer. I have not gone through any paper of using biostimulant. If any of the audience knows, I think they can answer this question.
Yeah.
Practicality, I'm just telling. So all the tissue culture is aseptic conditions. Biostimulant is usually they're not highly purified thing. A lot of complex material will be there. So unless you purify and then put it on the system, it might be a challenging thing. So maybe you can.
See, recently there are some studies where they have used P indica in tissue culture also with good benefit.
Yeah, it's yeah. Extract.
It was identified somewhere in the desert in Rajasthan. Now it's quite famous actually.
Yeah, Vishwa. One particular part where you showed that some of the ABA action inhibitors usage in strawberry, because strawberry is a non-climatic fruit. So its ripening mechanism itself is totally different. It is nothing, it is not responsive to ethylene, but it is responsive to ABA action. So their ABA action inhibitors, which you have used. So can we use something commercially to reduce or delay the ripening of strawberry? Because that is one of the very important problems in strawberry ripening.
So I don't have any answer for that one. If any chemical is available, that can be used, because it is not tested. Only they have used by RNA approach of ABA receptor. They have shown that it is involved in the ripening, but no chemical was tested on them. These chemical, none of the chemical were tested on the strawberry by using overexpression of abscisic acid receptor and by RNA inhibition of abscisic acid receptor. They have shown that abscisic acid receptor is promoting the strawberry ripening. And even in many other crops, like in mango, the color development and flavor development, it is regulated by abscisic acid. So, but not many studies are there to use of the chemicals. Only when we use this chemical, a lot of studies will be required, as otherwise, especially when we are using post harvest. the safety and residue, those things need to be studied. But first we have to identify some chemical that I have not come across anybody has tried any chemical which is related to ABA signaling in the strawberry to regulate the ripening process. Maybe your institute can try some.
Sir, another question that arises in my mind is that the increase of bioengineering, it's like research will be going on also. So are your researches incorporated with the bioengineering? So will it be efficient in your research?
Bioengineering means?
Like with the help of genetics and biotechnology,
Okay.
like are we doing ABA with the help of it?
Okay, ABA engineering ABA signaling pathway. So we have engineered the ABA receptors in rice to alter the stress response, and we have shown that some of the receptor they can increase, like we have one receptor PIL6, we can provide tolerant to salinity, drought, and cold stress. Some receptor they give specific tolerance to only to certain stresses. And another study from J.K. Zhu's lab, they have developed mutants of these receptors, because when abscisic acid signal is active, the stomatal closure is expected. Some control over stomatal closure is expected. But if you are not having any problem of water, you need not control the stomato. So in that case, they have shown that mutation in 3 of the ABA receptor, it actually increases the yield under field condition, because there is no control on the stomata. Stomata is always open, and it produces more photosynthesis. And the other point is like the pyrobactin mandy protein I told, which is an orthogonal receptor engineered. That is, by use site-directed mutagenesis, you modify the protein, and then they overexpressed in the plant. Now you have the genome editing technologies available. You can maybe modify one of those 14 receptors that are present in the plant, and you can use off-the-shelf agrochemical. You need not worry, because if you use the earlier approach of transgenic, getting approval and then environment release is a problem. So if you use genome editing approach, still you can get the same result, and you need not worry about all the regulation. It will be considered as normal plant. Definitely use of genetic engineering, I believe, although the initial cost is high, when the product is developed, this technology is easy to adopt by the farmer, because you are delivering this technology through the seeds. Farmer need not worry about applying anything. But all other technologies, farmer should have more knowledge. That is, he should know when to apply, where to apply, how much to apply, and then also the input cost in addition to the seed cost. So first approach, if we can deliver through seed, we should deliver. Then if not, then we can deliver through the production technology. Great question.
Thank you, sir.
Thank you.
Very good. Panbalagi and our team, Ashutosh, they're all undergraduate students. I'm really happy about that.
Excellent
Good.
question. Yeah.
Very good. And you have a good question. The only thing that you need now is air conditioner and other things.
Yeah.
Good. I mean, in the interest of time, Nataraj, you have a question? The answer that you elicit from him should be yes or no. Go ahead.
Good, Vishwa, very good, very nice, informative talk, especially concerning towards entrepreneurship development. I was just wondering, like in Italy, we have system 1, system 2 response for 5 different receptors. Do you have similar kind of information being generated for 14 different receptors?
In 14 different receptor, actually the concentration of ABA with which they are activated, only that is different. Still, all the studies are like several studies has to be done, which has not been done. Like, because ABA regulate the seed germination. In seed germination, the pile 1, pile 2, pile 3, pile 4, only these 4 are sufficient to regulate seed germination. Other receptors are not sufficient. But for each ABA receptor, we don't know whether out of the 14, how many are involved. Then there are combinations, are also multiple combinations. 14 ABA receptors are there, 9 protein phosphatase 2C into 3 SNRK kinases. It makes so many combinations, and then finally the target protein. So it is the number of combination is many, but definitely it is worth elucidating the specific process in which these ABA receptors are involved. Otherwise, classification is like similar classification is not there. All these ABA receptors, they are present in cytosol, and they can interact with the the transcription factor, and they can do multiple things. A lot of work is there for us to do.
Thank you. Thank you, Vishwa.
Okay. Yeah, we can discuss during the lunch time. I think already we are late.
Thank you, sir, for your insightful presentation. Now I invite Dr. Sheshashai, Professor and University Head Department of Crop Physiology, to come forward and present a memento to Dr. Vishwanath. Thank you, sir.
Thank you, Vishwa. Before we start, and Akshata invites Preeti to introduce the speaker, we just wanted to say this as a part of our tribute or contributions to the memory of Professor Uday Kumar. The alumni had contributed handsomely, and we deposited quite a bit of money with the Indian Society for Plant Physiology 3 years ago. And from the previous year, from last year, the ISPP has instated a Young Scientist Award for the best young scientist who does it in India, and one award is given to that person. Last year, it was Dr. Ramegowda, and this year we have the speaker, and we invite every year the recipient of that award. I wanted to thank the alumni for having contributed for this cause. Thank you, and over to you.
Now we have an invited talk by Dr. Soham Ray from IIRI New Delhi. I request Dr. Preeti NV, Inspire Faculty of Crop Physiology, to introduce the speaker.
Good afternoon, everyone. It's my pleasure to welcome Dr. Soham Ray, Senior Scientist at IIRI. Dr. Ray completed his PhD from IIRI and was awarded prestigious Inspire Fellowship. His research primarily focused on identification, cloning, and regulation of genes related to rice blast pathogen. As a scientist, he continued to explore plant stress responses at physiological and molecular level. He has published many articles in renowned journals, and he holds 2 patents. Last year, as Dr. Shesha said, he was honored with Professor M. Uday Kumar Memorial Young Scientist Award. Dr. Soham Ray, I welcome you to the dais. We are very excited to hear from you.
Hello. Yeah. So thank you very much. And I also like to thank the Alumni Association, Crop Physiology, for giving me the chance to present my work. And I also beg your pardon because the theme of the concept and the work I am presenting doesn't match. But this is the area I work, and this is the area I have not worked at all. So for next 15-20 minutes, you have to bear with me. So, okay. So, okay. So at the outset, I would like to, this is my tribute towards Professor M. Uday Kumar. Although means I am the awardee of this, but unfortunately I never had the chance to meet him. So, but I have heard about his work, and my namaste to him. So now, if I continue to pay tribute, I should also pay my tribute to my institute, IRI, from where I have done my MSc and PhD, and now I am scientist here. but the road was not the straight way. So after completing my PhD, I joined NRI Cuttack, worked there for 5 years. Then I moved to Kraizab Barakpur, worked there for 3 years. And then I came back to IRI. So in terms of physics, I have not worked at all, but luckily I am not a physicist. So I am basically a student of molecular biology and biotechnology. I have done my MSc and PhD in this subject. And my ARS discipline is also agricultural biotechnology. And when I was posted at IRI, I was placed at Agriculture Innovation Center. So this is the current status of our Agriculture Innovation Center. It is under construction. This current situation. When I joined for 3 years back, it was only on paper, and Vishwanathan sir was the in charge of that facility. And being the head of physiology that time, he asked me to join to plant physiology division, and that is my association with physiology, started my association with physiology. So when you are in Innovation Center at IRI and working with Vishwanathan sir, so what you have to work with? More obviously, the most recent innovation that is in molecular biology, that is genome editing. So this is the area I am working. So genome editing, as you know, it is the specific changes that is made in the genome, specific and desired changes at specific sites. And thanks to these heroes, due to their work, maybe 15 years back, which was a dream, has now become so household thing that the definition comes in Encyclopedia Britannica. Okay, so this area we are working with, and just to briefly introduce how we what the technology is and how we do it. That is, say this is a plant cell and this is the target in the genome, and you want to modify it. So what to do? You have to put a editor plasmid inside. Use your best approach, what you use, either agrobacterium or particle bombardment or chloroplast, whatever it is. You put an editor plasmid inside, and in this editor plasmid has two transcription system working simultaneously. One driven by RNA polymerase II. It encodes the Cas protein and along with the nuclear localization signal, and it has also a type 3 RNA polymerase which encodes a single guide RNA. So this encodes the Cas9 protein, so transcribed and translated, and then here it also encodes the sgRNA, and since it has a nuclear localization signal, it is transported back to the nucleus, loaded by the sgRNA, and this sgRNA guides it to the target site, and then it mediates the cut, and once it is cut, it is the cellular repair system which repair it, and in this process we get the mutations. Now this is for a single site. If you have multiple sites, what to do? Can you do it? Yes, we can do it. What you need to produce multiple sgRNA which can be individually loaded to the Cas9 enzyme, and simultaneously it can target multiple sites. So this is the area I am working with, multiplexing of CRISPR. And there are several technologies which are used for multiplexing. I will only discuss this one because this is what I am working with. So this is a patented process developed by Y. Yang and his group. So if we see the natural cellular system, our tRNA are produced as a polycistronic transcript, and it is processed in the cell. So they have utilized this strategy. So every sgRNA, the encoding unit is preceded by a tRNA encoding molecule. So you get a polycistronic transcript where every sgRNA is preceded by a tRNA molecule. And then the cellular RNases, there are two RNases, RNSP and RNSZ. They process it individually from here. You can see the scissors. So individual sgRNA are produced and they are loaded by the Cas9, individual Cas9, and then they can target to the specific sites. Okay, so this is the whole thing. Now, let me tell you, so this is not coming. So they say, for example, you want to multiplex 3 targets. You want to knock out 3 targets at a time. So you have to design a PTG. It is called polycystic tRNA gRNA assembly. So it is a 3x PTG. So you have 3 independent units. Okay, so to assemble produce this PTG, you need several stepwise processes, including primary PCR, overlap extension PCR, and golden gate. I am not going into the details, but trust me, it is bit complex, and basically the primer designing, because every primer is related to each other. Okay, so primer, and you need for 3 you need 14. If you are multiplexing, if you are targeting more than 3 units, okay, so there is a 4n+2 rule. So if you want to knock out 6 different targets, so you need a 6x PTG, and that needs 26 primer pairs, 26 primers, and then again you have to clone this particular PTG in this cloning site, and that is a directional cloning. So every primer is related, and it is it's a bit complex to design manually, and no software is there to support it. So we have developed a software that helps you in this process. It takes very simple inputs, the guide RNAs, and little more inputs, which you will get in the help of this software, and it gives all the required outputs. And recently we have got copyright of this software, and we have also validated this software, output of the software, through several wet lab experiments. Now, I will give you one example of such a wet lab validation, and in the process, I will also demonstrate the work we are carrying out in our lab. So, this is we are working in a with a protein family, gene family, that is called matrix metalloproteinase. Now, these matrix metalloproteinases are very well known in case of vertebrates. Okay, what they do, they are, as the name suggests, they are they perform their work in the matrix, and these are basically the collagenases. So they degrade the collagen of the matrix and helps in the movement of the cell. That is called metastasis. So metastatic movement of the cell, they help. So in the process of aging, cancer development, everything, this MMP has a role. But interestingly, plant also have this gene family, but plant do not have collagen. So what this particular group of genes, gene family is doing in the plant? So that was a question. Okay, before that, I should also tell that these are encoded, these are synthesized in the ribosome associated with rough endoplasmic reticulum, and once they are synthesized, they go to the secretory process, but they don't secrete outside the cell because they are anchored to the plasma membrane through a GPI anchor. So it is something like that in the plant cell. If you see, so this is this cell wall area. So here it is attached to the cell membrane and displayed in this portion, this matrix portion. And if we go to the structure, it is synthesized as a pre-propeptide, and here it is present as a propeptide, that is inactive protein. When this portion gets cleaved off, it is this has to be gone off, then only it becomes activated. Okay, so but the question still is what this kind of protein are doing inside the plant, because plant don't have collagen. So we are trying to understand this process. But before that, to understand this process, we have decided that we will use rice as a model system. And we try to identify how many MMPs are there in the rice plant. So they have some, these are some typical features, and star marked are the critical features of the this MMP proteins, and this green star indicates plant specific MMP. So based on this criteria, we have developed a pipeline to identify the MMP candidates. And using this pipeline, we have identified there are 3 classical candidates, candidate MMP in rice. And we have also seen through in silico expression analysis that these are differentially expressed under different stresses, also in case of development, but also in case of stresses. And they are present in the apoplastic space. They have pleiotropic roles, but exact biological function is unknown. So to understand the biological function, we have decided to follow this pathway. So we have to develop knockout and overexpression lines for this. And to develop this, today I will tell the story of this knockout line generation only. So we have used our software. So these are the three targets. Every MMP gene we have decided to knock out. We have to use 2 guide RNAs to introduce insertion deletion, and in the process that the genes will be knocked out. So we have multiplexed. We have decided that these 6 guide RNAs we have selected for knockout purpose, and we have decided that we will use a 6x PTG to perform the multiplex editing. Okay, so using our software, we have designed this 6x PTG, and this is the designing process. So you need to generate this T1, this T units at the tRNA units, this G units at the gRNA scaffolds, and then you have, through overlap extension PCR, you have to join this TG units, T units and G units to make TG units. So for 6, you have 6 TG units, and then using Golden Gate assembly, you have to join all the 6 TG units to make a 6x PTG, and then you have to clone it into the editor plasmid. And once we have done that, we have sequenced the editor that portion of the editor plasmid, the MCS of this editor plasmid, and check the sequences, and we have found that all the whole PTG assembly has been developed perfectly. So this shows that our PTG, this software is doing fine. Then we have developed plants, transgenic first you have to develop transgenic plants, putting the editor plasmid inside. So these are the transgenic plants generated, and then we screen those plants using the first we have screened with Cas9 specific primer pairs. So appearance of this 912 base pair band says that it is a transgenic. That means the editor plasmid is inside. So these are the positive plants, and once we got the positive plants, and we have sequenced these this MMP 1, 2, and 3 gene target regions. We have amplified and given for Sanger sequencing. Okay, so this is the, once we got the result, this is the result of our sequencing results, I will say. So we have found that in case of MMP1, this is only in case of MMP1. So we had 2 targets, sgRNA1 and sgRNA2. So, and if you see in this PTG construct, this blue one at the MMPs means this blue guide RNAs are actually targeting the MMP1. So you see physically in PTG it is apart. This is the guide RNA1 and this is the guide RNA2. This is guide RNA1 and guide RNA2. So that means, and we found that these are the different alleles we have produced. So in guide RNA1 region also and guide RNA2 region also, we have got mutations. So this says that our processing is proper. So our software is working properly, and few of them you can see that these are the knockout probable knockout mutants. But unfortunately, we could not get any mutants for MMP2 and 3. So what might be the reason? The reason is probably these are very resistant to mutation, because we have analyzed the 3000 transgenome sequence available from ED database and found that there are very low number of mutations in the coding region, and whatever mutations are there, either they are synonymous or they are conserved missense mutations. So probably it do not tolerate the mutations. And even we have studied the MMP this sequence of this MMP genes in wild rices also, and found that there are very low deviations from the conserved sequence. So most probably these are very resistant to mutation, but still we need to generate if knockout is not possible, we will try to generate knockdown through RNAi. And we have also produced the overexpression plants. So these are the southern blots. We have got some single copy transgenics. So here my work stands now. So this is what we have selected AMP1 mutants and overexpression lines for all this. So this portion is still remaining. So sorry, I am not showing any physiological work because it is still pending. So maybe next time, if I get the chance to present, I will present something on physiology. So thank you very much. But I would like to acknowledge the invitation, this the IRI means alumni. Let's go. So Alumni Association Crop Physiology, I would like to thank them for calling me. I will also thank my institute to give me the permission to come here. In fact, our JDR is here only. So, I would like to thank Vishwanathan sir. He is not only the mentor for us, he is mentor of genome editing for whole India now, but we get little bit extra time from him because of physical proximity. These are my collaborators, and these are my staffs working with me. And last but not the least, I want to thank everybody who are present here. Thank you very much.
Hello, sir.
Yeah.
I am Ankit, so
Yes.
PhD scholar, Department of Plant Biotechnology.
Yeah.
So my question is, so you have said that the other two MMP 2 and 3, so which is very resistant to mutations, but your approach is on the CRISPR approach.
Yeah.
So how it is related to your wild? You have studied with the wild nature, so it is resistant. But now you are specifically targeting those MMP 2 and 3. So what is the reason for that exactly?
See, sometimes the genes are conserved because of the very nature that they are vital. Means if you knock that out, the plant cannot survive. That might be one reason, but I am not sure for that also. But I can give you the reference that for all, not only MMP 2 and 3, also for MMP 1. So in rice, you get only one paper about MMP 1 knockdown, not even knockout. And there are no other references on MMP 2 and 3 for knockdown or knockout. So it might be, there is a possibility, but still we are in the process of investigating it. If we get, that is fine. If we don't get, then we have to think other ways to prove that these are really vital gene, or without that the plant cannot survive, or whatever it is.
Okay, sir. So I'll just to add on that. So whether it is really the Cas9 or some modifying protein which is entering that region, that is difficult, or like targeting the specific guide RNA which is making difficult.
See, this is there is no modifying enzyme, but you are right. Means Cas9 has to get access of the target. That is one thing.
Yes.
If it is if it is in heterochromatic region, or at that point of time when we are trying to develop the mutant, okay, that particular moment, if it is not accessible to Cas9 enzyme, even if your guide RNA is proper, then also it will don't produce mutant. Okay, so that might also be a reason.
Thank you, sir.
Nice presentation and wonderful work, Dr. Soham. I have one small technical question. So how many guide RNAs can you put theoretically in editing construct that Cas9 can handle? And then what are the chances of those, again, theoretically being edited? At least from the literature.
So see, there are different reports, but the paper which I was referring, that is a landmark paper. You can see that it has got almost 14,000 citations. Okay, so 1400 citations. So what they suggest, they have only patented this technology. What they suggest that up to 6 it works well. Beyond 6 it doesn't work that well. But now people you are using different strategy. And recently I have read one paper. They have said that they have multiplex 32 simultaneously. Okay, but yes, more you multiplex, difficult to analyze. So
What are the chances of you getting the edits in the same plant for some of those?
this is a completely probability
chance
game.
factor.
Completely probability game. So all are independent events, you multiply the probability. So so you can understand it will be very less.
Thank you.
Thank you. Yes, sir.
Just to continue with that. So when you do this multi-locus genome editing, is it by choice you want to do for multi-family genes, or it can be for any of those?
Anyone, sir.
So if I go by three or four, what does literature say in terms of what is the probability of you getting all 4 knockouts? Can you get partial knockouts? Only 3 out of the 4, only 3 working?
Yes, sir.
So
mostly you will get single. Getting double is again a probability factor. So all are independent events. And you see, there are means first you will have to get transgenic. That has a probability. In transgenic, you have to get mutation. That is again a probability and independent event. So multiplication.
So your
every time.
So your objective is not to get a phenotype with is knocked out by multiple genes.
Yeah,
it is more of getting the independent
yes,
events of
sir.
those.
I was expecting when I started this work, I was very novice in this work. 2020 only I started this. So my plan was, if I get multiple mutation at a time, it's fine. If not, even if I get individual, I will cross it and make it, bring it in the same background. So sorry, I rushed a bit because it was already late.
Thank you, sir, for an informative talk. Now I invite Dr. Nataraja Karba, Professor and Head, Department of Crop Physiology, to kindly come forward and present a memento to Dr. Ray. Thank you, sir. Ladies and gentlemen, now we will take a break for lunch. So we are already late. So lunch will be served in the UG Boys Hostel right behind the auditorium. So after the lunch, we have an interesting session, that is the panel discussion on entrepreneurship development. We have 3 successful entrepreneurs and a venture capitalist, and it is going to be very interesting and it is very essential and important for us. So please get back to the hall after the lunch quickly. Thank you.
I think 45 minutes should be good enough for 30 minutes. Okay, so I heard from Dr. Shesha 30 minutes. Please do come back by 2:30. So UG hostel is right behind this building, so you don't have to go too far. Okay, please do come back by 2:30.
ಅವ್ನಿಗೆ ಹೇಳ್ತೀನಿ ಒಡ್ಡಾಗ್ ಕೊಡ್ಬೇಕು ಅವ್ನಿಗೆ. ಸ್ಕ್ರಿಬ್ಲಿಂಗ್ ಪ್ಯಾಡ್ ತಗೊಬಂದಲ್ಲ, ಸ್ಕ್ರಿಬ್ಲಿಂಗ್ ಪ್ಯಾಡ್. ಹೆಣ್ಮಕ್ಳ ಜೊತೆ ಊಟ ಮಾಡ್ತಾ ಕೂತವನು ಸಂದೀಪ. ಪ್ರೋಗ್ರಾಮ್ ಶೀಟ್ ಇರ್ಬೇಕು, ಯಾರ್ಯಾರು ಏನಿಲ್ಲ ಇನ್ನೊಂದಿಲ್ಲ. ಶೇಷ ಕೊಡ್ಬೇಕು, ಅವ್ರು ನಾಡ್ತಾನಲ್ವಾ, ಶೇಷ ಕೊಡ್ಬೇಕು, ಇದಾಗಲ್ಲ. ಈ ಪ್ರಿಂಟ್ ತೆಗಿಯಕ್ಕಾಗತ್ತಾ? ಹೀಗೆ ಅವ್ರ ನೇಮ್ ಇದಾಗುತ್ತಾ? ಸ್ಕ್ರಿಬ್ಲಿಂಗ್ ಪ್ಯಾಡು, ಪೆನ್ನು, ನಾವು ಅವನ್ ಸಂದೀಪ್ ಕೇಳ ಗೂಗ ಅವನು ಗದ್ದು ಮುಚ್ಚಿ ಆಟ ಆಡ್ತಾರೆ, ಯಾರು ಎತ್ಕೋಗ್ಬಿಡ್ತಾರೆ. ಒಂದು 5 ಸ್ಕ್ರಿಬ್ಲಿಂಗ್ ಪ್ಯಾಡು, 5 ಪೆನ್ನು. ಪೆನ್ ಇಲ್ಲ, ಪೆನ್ಸಿಲ್. ಈಗ ಪ್ಯಾನಲ್ ಡಿಸ್ಕಷನ್ ಇದೆ ಬನ್ನಿ ಸರ್. ಇಲ್ಲ ಈಗ ಸ್ಟಾರ್ಟ್, ಈಗ ಸ್ಟಾರ್ಟ್. ಬನ್ನಿ ಊಟ ಮಾಡಿದ್ರ? ಊಟ ನೋಡಿ ಈಗ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಬರೋಕ್ಕಿಂತ ಬನ್ನಿ. ಊಟ ಆಗಿಲ್ಲ ಅಂತಂದ್ರೆ ಹಾಸ್ಟೆಲ್ ಊಟ ಮಾಡಿ ಬನ್ನಿ. ಹೌದಾ? ನೋಡಿ ಯೋಚನೆ ಮಾಡಿ. ನಾನು ಆಗ್ಲೇ ಇದ್ದೀನಿ. ಈಗ 2:45 ಸ್ಟಾರ್ಟ್ ಆಗೋದಿತ್ತು, ಇನ್ನೊಂದು 10 ನಿಮಿಷ ಅಂತ ಹೇಳಿ. ಬಾಟಲ್ಲಿ ಎಲ್ಲ ಊಟಕ್ಕೆ ಹೋಗೋರು ಹಾಸ್ಟೆಲ್ಗೆ ವಿದ್ಯಾರ್ಥಿಗೆ ಇನ್ನೊಂದು 5 ನಿಮಿಷ 10 ನಿಮಿಷ ಅಂತ 10 ನಿಮಿಷ ಬರ್ತಾರೆ ಒಳಗೆ. ಎರಡು ಮೈಕ್ ಕೊಡಿ ಅಣ್ಣ. ಇದು ಇಲ್ಲೇನಣ್ಣ ಸರ್? ಸರ್ ಈ ಮೈಕ್ ಸರ್, ಈ ಮೈಕ್ ನಲ್ಲಿ ಮೂವ್ ಮಾಡ್ಬೇಕು. ಅಲ್ಲೇ ಎಷ್ಟಿದೆ? 4 ಇದೆನಾ? ಹಾ, ನೀರಲ್ಲಿ ಮೂವ್ಬೇಕು ಇಲ್ಲ. ಟೇಬಲ್ ಒಂದೊಂದು ಕ್ವಶನ್ ಇದನ್ನ ಬೇಕಾದ್ರೆ ಮೂವ್ ಮಾಡೋಣ ಅಲ್ಲಿ ಅಲ್ಲ ಬಿಚ್ಕೊಬಿಟ್ಟು ಶೇಷ ಅವ್ರ ಕೈ ಕೊಟ್ಬಿಡ್ಲಾ ಇದನ್ನ ಇಲ್ಲಿ ಕೂತ್ಕೊಂಡು ಮಾತಾಡ್ತಾರೆ ಶೇಷ ಬಿಚ್ಬಿಡು
ಮೈಕ್ ನಾನು
ಹಲೋ
Good afternoon. I hope you had a some sumptuous, satisfying lunch. Yes or no? Ah, no. Silence means they're sleeping. I did say that eat well, but don't sleep. Okay. So before we start the most important session of the day, I wanted to introduce to you a very, very important person who has come. And may I have the pleasure of calling Dr. Madan Pal ji for a minute on the stage. He's been the backbone, ribs, head, brain of Indian Society for Plant Physiology for quite a long time. And I wish he will continue like this forever. Because there can be no better person than him. And I hope Vishwa agrees with me. Yeah. So great pleasure in welcoming him. Give him the flower for this event. And I will invite myself to give him this plaque. Thank you very much, Madan Palji. And society, as I said, has been extremely supportive and encouraging. That's what is more important, giving us critical suggestions and advice whenever it is required. It's a marvelous association that we have been having with the Indian Society for Plant Physiology. Thank you very much for both Vishwa and Madan Palji for gracing today. And it's something which is very, very important for me that I'll cherish for this forever. Thank you so much. And for the afternoon session of the panel discussion, I'll ask Akshata to go ahead.
Welcome back, everyone. Now we move into one of the most important sessions, that is panel discussion on entrepreneurship development. I welcome Dr. Sheshashai to moderate the panel discussion. Welcome, sir. Over to you.
Over to me. Oh, I don't need this. So good afternoon again. And this one of the most important things that at least I feel is the next thing to come is, what should I do? No, but it's coming like this. Okay. You can hear me? Wow. Okay. So one other thing that is expected, which everyone feels, is that either you get a job or you give employment to others. How many of you want to give employment to others? Raise your hand. That's all? We all? Or how many of you want a job? Okay. So you want a job or you want the job? The job, you have to work very hard, isn't it? A job, you will get bus conductor, driver, cleaner, whatever. So anyway, all the very best with whatever that you have in your minds. But what we have now is the most important thing is every one of us should become entrepreneurs. And I used to call it entrepreneurs. I believe it is entrepreneur. All right. So this entrepreneurship is something which requires a lot of courage, which I honestly don't have. Praveen and Shailaja, we've been talking about this kind of courage. We require someone who has the kind of correct guts, which are made of wire in the stomach. So let's invite three of the very successful startups and who have done exceptionally well globally. And let me have the pleasure of inviting Praveen Vemula, who is a senior scientist in STEM, Bangalore. Huh? Oh, yeah. Why don't you give him a flower? And at the same time, in our NCBS CCAM association that we have, where the startup ecosystems exist, we have one very close to heart a startup which is called C6 Energy. C6 Energy, you will listen more from Shailaja Noori, who is the co-founder and managing director of that company. The great affection that we have is that they started this with the advice which they actually interacted with Professor Uday Kumar in the beginning, and they are huge now. And I'm glad some of our students are working with your company, in your company. I'm very happy. And I believe someone got the offer yesterday. Yeah? Okay. So that's one thing. And of course, this is something which is very special, emotional, and very important for us for the Department of Crop Physiology. Gopal Krishna, who is an alumni of our department, who did his master's with us, PhD with us. Then he did his postdoc in Yale University in the United States. He was a perfect molecular biologist. And now he was one of those very early persons who asked very horrible questions. I remember this once when we were talking to them when they were in undergraduate program. Why ATP is called ATPase? Why is it a phosphatase? It should be a kinase. It adds phosphorus to ADP and makes it a phosphorus triphosphate. Oh my, I didn't sleep that night, I'm sure. Whenever I think about ATP, I always remember Gopi. Then I realized and I read it is called reverse ATPase. Thanks, Gopi, for that. And Gopi has started a company called Theracus, and it's a service providing company for all the genomic support. And mostly he works with medical sciences, but I'm sure DNA remains very much the same chemical, whether it is taken from a donkey, human, or a plant. So let's see if we can check what is it that we have with them and all that. And the most important thing that we require for all these business modules to sort of function is money. And that's all. That is what it is. When money talks, they say bullshit walks. So it's money that is very, very important. And the problem is that we don't get it. It's all there everywhere. Water everywhere, but no water to drink. Now that's the situation. Means there are certain questions that are asked by the funders or the people who can support this kind of startup ecosystems. What are the questions that they ask? Do we have an answer? You may have a brilliant idea, but does it have a potential to get converted into a business module is the most important question. So I have great pleasure in inviting Aman from venture capitals called the Ankur Capitals. And I'm glad he is here with us. So what we do for the next half an hour or 1 hour is that we listen to each of them as to how did they get this. Sometimes I'm sure these ideas were most of us think that these ideas are actually silly. I remember this once somebody went and asked his father that apple fell from the tree. Why did it fall? Father said, don't worry, go take care of the sheep because the apple always falls. It doesn't go up. Yes, that's the question. Why it did not go up? Why did it fall down? So we know what is gravity now. But Newton did not know before he discovered that there was gravity, but he had asked that question. Why did it fall? So therefore, this question was a stupid question, silly. But then when you really analyze this, I mean, that has all sorts of science that we have now developed depends on the gravitational forces that we have, satellites and drones and whatnot. So we have all of them here. So let's go and listen to Praveen. Praveen is a chemical engineer by training and is a passionate medical science biologist. Can you believe this? So this transition, agriculture universities, I think we should know this. Blinkered visions are important, but then the mind should think of all sorts of things. So he has made something which are important for agriculture. And his product that we talk about is called Kisan Kavach. And that has become really, really famous. And I'm glad this year in the month of March, his paper was published in Nature Biotechnology about the concept that he has developed for protecting farmers from the pesticide, pesticide that when they spray this pesticides, it can cause hazardous reactions on human skin. And other than that, he has a lot of other kind of startup ventures that he has across the globe, I should say, not just in Bangalore. So let's listen to Praveen for some time. And I think we will take about 10 to 12 minutes each, Praveen. And then we will have, please be open. This is a very beautiful opportunity for us to ask questions. Whatever may be that, don't worry whether the question is silly. All right? Thank you, Praveen.
Thank you so much. This is going to be a little impromptu presentation. So earlier we thought of just talking, then can I have the clicker, please? And this is also side effect of calling professors for panelists. Okay, we'll tend to talk. So morning session, you heard a lot about strategies to protect the crop, right? So on the contrary, we are focused on protecting farmers. I'll tell you how we have been developing technologies to protect the farmers. And I'm coming from the neighbor institute called INSTEM. Yeah. Great. Thanks. So, you all know, all these pesticides are highly neurotoxins, right? They are designed to kill the pests. Typically, how they work is we have this enzyme called acetylcholinesterase, which plays major role in all your neurological function, neuromuscular function. And our pesticides are designed to go and bind them covalently, irreversibly, so that it can inhibit them. So when you have this quantitative inhibition, that leads to severe neurotoxicity, paralysis, and death. That's how all the pesticides die when you spray them, right? So, but unfortunately, they don't discriminate between pest and human because we have same enzyme, same function. So when you get exposed, we get the similar toxicity. So typically, farmers in India and developing countries, can you just play that? So they spray pesticide without having any protection. It is almost like walking under the poisonous threat. It falls on their skin, they inhale and so on. But right now, if you see, there are absolutely no protective gear to prevent them. So then we thought, can we develop a protective gear for them to prevent the exposure? But we wanted to see how much toxic effect they have. As a classical scientist, what we see is when you think out the problem, you go to the conventional literature and read them. And interestingly, all the conventional literature, what it says is people have to get exposed for a decade or so, then they start showing the chronic symptoms, which is completely we couldn't comprehend that fact because these are highly neurotoxins. How could they not have any acute effects? So we kept the your scientist mode off and took the entrepreneur mode on to go and identify the problem for the root cause. So that's where we launched a survey. Almost 8 months we spent in the field, and almost 60 villages in Telangana we visited, 200 farmers we spoke. They're also not randomly talking. For each farmer, we used to time in such a way that when they spray, you go and spend 2 days with them to see what kind of pesticide they use and what kind of exposure they have. Do they have any side effects? What kind of protective measure they use? I think that is one of the best decision we have made that completely changed the way we think about pesticide toxicity, because almost all of them, when they spray, acute toxicity they have like within same day, within hours. They suffer with like a vomiting, diarrhea, muscle pain, breathing disorders. If a higher concentration got exposure, that leads to loss of vision and death and so on. So which shows the problem, the intensity of the problem that exists, and absolutely there is no technology to protect them. So what we thought is then, can we develop a dermal gel, which can then apply like just a dermal cream, which can prevent them going through the skin. So then what we have done is like physical barrier gels, they don't work because these are the chemical designed to penetrate skin much faster, right? That's how they kill the pest. So that's why we came up with this concept called like, reactive gel. That means like this not only physical barrier, but is as a chemical reaction. It attacks any pesticide comes in contact, chemically deactivate in such a way that it can no longer inhibit the enzyme. So in that way, we can completely prevent the pesticide-induced toxicity. So again, if you see like it's a completely unorganized sector, you see like hundreds of pesticides on the market, right? So you need to have one technology which can deactivate all of them. So, but if you see like all of them, they share one interesting chemical feature called activated esters, right? That's a more activated is the more toxicity. So what we thought, use the fundamental hydrolysis reaction. And if you take this nucleophile mediated hydrolysis, convert this ester into an acid, which can no longer inhibit the enzyme. So for that, like again, like we designed this gel, just to simplify that, like we have this chemical moiety which act like a scissor and connect to the polymer. And by using appropriate components, you can make them as a oxime gel. As a control, you can take a physical barrier gel, which act as a physical barrier, but doesn't have the ability to break down the pesticide, right? So once we have that, like you can go and expose to the animals, like for example, 3 group of animals, you can take and expose them either directly or through just barrier gel or active gel. The way you measure the exposure to the pesticide is like you collect the blood and the tissues and measure the enzyme activity before and after, right? So the level of exposure directly proportional to the inhibition. So if you see here, like, when you have this exposed either directly or through physical barrier gel, you see significant drop of enzyme activity. Whereas when you have the active gel, you don't see such a drop, which clearly suggesting that they are able to deactivate, degrade the pesticide before it passed through that. Then we have done extensive studies to study their neurotoxicity and all of that. Then we are super excited. Then we thought like, we will go and develop this technology further. So to translate that again, like we went and formed a spin-off company from Instem called Sepio Health. And through that, like we developed formulations for human dermal applications. We have done extensive safety studies and so on. But like, again, as a scientist, if we think it's technology working very well, right? But at the same time, we need to keep our inventor bias aside, see from the user point of view, whether does it work, the adaptability is there. Then we can clearly see 2 problems, right? One is the cost, because they have to keep reapplying every time they spray. So that means you have to repurchase, and we are targeting the farmers who have purchase power is very low. So the cost would be an issue. And the compliance, second one is because every time all over the body, they have to apply whenever they spray, right? Maybe they might not be, everyone might be like, comfortable to do that. So, but again, instead of reimagining the problems, so we thought like, get the actual user feedback. The best part of like staying as a neighbor to GKVK is like when come to Krish Mela, we used to set up stalls and talk to like, hundreds of farmers and displayed this technology, and we got the same feedback like what we anticipated. So that made us to think about alternative technologies to protect them. If you see these farmers, many of them, they think that wearing these coats and all, like a full sleeves and all, they protect them. They wear full sleeves, they wrap around towels and so on. But it actually has the negative impact. It can absorb more and act like a barrier, like a reservoir. So, but imagine now if you have a fabric, instead of a simple fabric, if this fabric also has the ability to break down the pesticide, then we might be able to solve this problem, right? So that's why, again, we designed another specific molecule. This is like a chemical scissor, which can be attached to the fabric. Now this activated fabric, you can stitch into any format you want, like, pant, shirt, and mask, and so on. So then again, we went and tested their ability to prevent that. Again, expose the animals like, through normal fabric, oxygen fabric. After that, if you collect the blood, and you can see normal fabric couldn't prevent the penetration of the pesticides, but whereas oxygen fabric completely prevented the exposure. And same thing, you can see the tissues as well. But like, farmers, if you see, they keep getting re-exposed, right? So we wanted to see whether if you have the re-exposure, will it still work and protect them? For that, again, you can take these animals, same animals, we keep re-exposing to the pesticides over 5 days, but on the same fabric without changing on the same fabric, you do that. When you do that, like, when you get re-exposure, they're completely, direct exposure or the normal fabric, you see 100% death. Within 4 days, all of them, they die. But when you have the active fabric, it completely protects them 100%, survival. You see, that shows that irrespective of how much ever exposure it has, it acts like a catalytic and cleave them, right? So if I take like 5 more minutes, so then we thought like, this is in a bulk exposure, right? But these are highly neurotoxic. Even small exposure can lead to severe neuronal damage internally, which most of the times we might not even realize. So wanted to see, can we prevent that as well? So one way to measure your neuronal damage is you measure the damage of sciatic nerve, because this controls your function of the limbs. So to measure that, you can take the animals, put like a different colored dyes on them and make them walk and collect the footprints. And you can measure the distance between the steps they are taking and the angles they are taking and so on. Put all those numbers in a complicated equation, can measure something called SFI. Okay. The thumb rule is when this number comes, when it is completely healthy animal, healthy neuronal system, this number always comes from +10 to -10. Okay. If anything, this number goes below -20, consider the severe damage. The more negative number is higher damage. If you see, when you have the direct exposure before, all of them in a healthy condition, but when you single dose exposure, it lead to like severe neuronal damage. And same thing when you expose through normal fabric also, you see them, right? But the same dose exposure, if you do through our oxime fabric, it completely prevents them. We're suggesting that even minute amount of the pesticide also not able to pass through this without getting deactivated. So another impacted factor is, like, as I mentioned, when they get exposed, they have this shivering and all, right? The reason is because once it goes and inhibit the enzyme, you see the overstimulation of the neuronal network. And we wanted to see whether it can prevent that. So this is the person, like, we never got exposed to pesticide. It's actually a graduate student who developed this. And if you measure the neuronal signaling here, you don't see any signal because there is no active movement. But same thing, if you measure to the farmer who got exposed to them, like you can see the all neurons are firing. Again, here, there is no voluntary movement, right? But it's completely involuntary because of the exposure. That's how, like, when they get exposed, like, they have this uncontrolled shivering and tremors. That's how it happens. So we wanted to see, can we prevent that as well? So for that, like, again, we can measure this into the animals. When you have non-exposure, you don't see any signal, but when you expose either directly normal fabric, you can see there's completely involuntary movements, but the same exposure with oxime fabric completely protects them, which is clearly suggesting that indeed this fabric has the ability to deactivate them completely before they're getting exposure. And these all are for the dermal exposure, right? Like a physical suit. But what about the aerosols? Because they can inhale as well. So to measure that, like, we build a box, like, where you can fill this box with the pesticide aerosols and make them breathe these animals either directly or through a mask made with the normal fabric or the activated fabric. Again, you can collect on tissue and see when you expose to oxime fabric, it's completely prevented the enzyme inhibition, which is clearly suggesting that this fabric can able to deactivate aerosol form of pesticides as well. So once we have that, now we went and test optimized in such a way that, because keeping completely user centric, because they work in the sunlight, it has to work in the sunlight, it has to be stable, and also it should work in a cold winters and hot summers, it should work all of them. So optimize all of that. With that, like, we built this suit called Kisan Kavach. So it has the, like, both the pant and shirt with a hoodie to protect the scalp, and also inbuilt mask, which can be that. And this is highly washable and reusable. Even 150 cycles you can wash, it still retains its activity. So easily they can wear for a couple of years. And this has been tested, like, beta test, many farmers are being using. This is Professor Arish from Horticulture College here in Agriculture University. And people from ICAR also, IH Horticulture Research also we've been using. So far, like, we have seen phenomenal feedback. So hopefully, like, I've shown that, like, how, like, identifying the problem, taking our journey from lab, whatever we develop, to the field and to the land, like, where right now, like, people are using, like, across the multiple states, and they see very instantaneous relief, because we selected the farmers, like, every time they used to spray, like, instantaneously, they used to feel completely body pains and vomitings and all. Now they have been using, like, multiple weeks now, like, absolutely don't feel any effects at all. So with that, like, I'll stop here, because right now, like, we are working with some of the government, like a Telangana government and all, to see whether they can purchase and distribute to the farmers. And also we are reaching out to a lot of companies as a corporate societal responsibility, CSR efforts, they can purchase and give to the farmers and so on. There are many individuals also, they're adopting, like, 100 farmers, 1,000 farmers, and donating them. By the way, just the cost of this is like a 3,000 rupees for suit, which can use for multiple years. So with that, I like to stop here. This is a fantastic group of people who developed from our lab this technology, and this is the funding source. I'll stop here. And thanks, sorry for taking a little longer time.
I think we will definitely have some questions. We will keep them for the discussions. At that time, we can come back to that. And I mean, I strongly feel that these kind of technologies are extremely useful for the farmer, and especially agriculture university. And I wonder why we cannot think about such kind of technology, because we did not study those scissors, which were some chemicals which will degrade another chemical. We never study those things in agricultural colleges. Maybe that's the reason why it stopped us from thinking about that. But it doesn't prevent me from collaborating with such people, right? Simple. I mean, what you do, you do. If you cannot do, collaborate. Simple. So, I mean, there we'll get back to them. Now, this entire concept that we developed this time was something called the elicitors and growth stimulants. I mean, we all have a desire to grow, but then we always need some stimulation. You agree or not? Whether it could be Koli Tata or whatever he did in the evening, some stimulations all of us need for sure. So that stimulation that can come from anywhere. That was a clever decision or clever idea that Sailaja and others had. Why not take it out from some of those things which are growing in that bloody ocean without anybody touching it, right? So let's listen to Sailaja as to how they got this idea of establishing a very flourishing company called the C6 Energy.
Hello, everybody. Good afternoon. I'm Shailaja, co-founder and chief scientific officer at C6 Energy. So today I want to share with you the journey we had over the last 14 years on how we built the company, what was involved in actually starting from where we were just after college to where we are today. So I would say we started as it was very accidental entrepreneurship, which we started about 14 years ago. And it started when we were working as a team for one of the competitions when we were all students at IIT Madras. And post that, we've also participated in Able Best, which was one of the entrepreneurship competition. And post that, in which we were working on some concepts on making biofuels from biomass and looking at replacing crude oil. And some of those concepts matured, and we decided to start C6 Energy in 2010 in the campus of IIT Madras. And the question we asked when we started C6 was, so today we are living on millions carbon, which is in the earth for millions of years ago. All the oil that we are using today, all the crude oil that we are using today is from plant and animal matter, which is buried like millions of years ago. So can we actually have a solution that is independent of land and fresh water, which can solve this problem at a global scale? And that was the question we were pondering on. And we realized that there are many aspects which we are dependent on crude oil for. For example, even a lot of agricultural inputs like urea is made from Haber's process, which requires energy, right? All the clothes that we are wearing, all the plastics that we are using, everything is derived. I mean, a lot of it is derived from fossil fuels. So our goal was, how can we address that portion of fossil fuel which is not replaceable by electricity or renewable energy? So that seems to be a very significant portion. It is about 10% or higher. And we said, how can we address that problem with something that is not dependent on land and fresh water? And when we did the calculations, we came to a conclusion that land-based biomass will not be able to solve the problem, because there's already a lot of pressure on arable land, agricultural fertility, and water requirement. So that's when we shifted to the sea. And we said ocean agriculture is definitely possible, because oceans, at least the tropical oceans, have the perfect temperature for plants to grow. And if we can utilize like a very small fraction of the area that is on the oceans and grow biomass, that can solve some of these major global problems. And that's how we started in 2010, with the vision to create a sustainable carbon negative feedstock to solve these major global problems, right? And so for us, the focus is on seaweed, which is at the center, which is a proprietary carbon negative feedstock. So today as a company, we work in two different divisions. One, looking at how we can grow this biomass at a very large scale on the oceans. And the other one, how do we convert this biomass to products which are sustainable and which can contribute to improving climate change and all of that. So some things that we found very interesting in tropical seaweed farming was this is a biomass which is grown all year round, compared to land-based biomass, which you have definitive cycles, and it has very high biomass productivity. So it appeared like a very interesting raw material for us to work on. And the main issues I'll talk both about upstream and downstream. The main issues in the upstream to grow this at a very large scale was, all of this is done in a very manual way in Indonesia and some parts of India. So there's been no innovation in the way it has been cultivated. So we have come in and we have actually focused on bringing in technology, both at a mechanization and machine levels, but also on a community level, where we are actually working with people, coastal fishermen, to grow this in a more sustainable manner. So some of these efforts culminated, and this year we actually launched a 1 square kilometer farm in Indonesia, which is completely mechanized, and which was a huge milestone for us as a company. And on the other hand, we wanted to understand what are the applications that we can make from a raw material, because obviously growing it at large scale is not the only problem. We also have to convert it into useful products. So we first started targeting with agri inputs. I think that is when in 2013, we first started interacting with Professor Nataraj Karba, Professor Uday Kumar, and we wanted to understand how some formulations that we can develop from seaweeds can actually influence crop growth and development. And we also have formulated other ideas or domains where we felt seaweed could actually contribute to solving some major challenges in food, in replacing single-use plastics, in decarbonizing the entire chemical industry, and ultimately fuels. So that's how we bucketed them. And in terms of downstream, we focused on how we can generate completely new active ingredients from seaweed, which can influence plant health, and which can also be developed into food and single-use plastic replacements, and also into chemicals and fuels. So we work on developing distinct platforms, technology platforms, to convert this. And so we have commercialized some of the products in plant health. So these are biostimulants. These improve plant growth and development. There's a lot of science that we have worked on and published. And these are again patented products which are being sold globally. So that is one of the product lines. So we have two products in that. One is on the biostimulants, which are used to improve plant growth and development, and the other line of products which are used to improve immunity in plants against viral diseases. So we also had a very long-standing collaboration with crop pathology department at UAS. So I think a lot of product development happens at interdisciplinary stage. I think this is a perfect example where people, companies like us, which have very minimal background in agriculture, can actually come and intervene and provide solutions and work with institutes like US, Bangalore, and other agricultural institutes to actually translate them into the field and provide solutions to the farmers. And in other verticals, we are in the scale up and research stage. So some of them, like bioplastics, we have shown proof of concept that seaweeds can be converted into like coatings, which can go into cardboards for providing grease resistance. We could also make prototypes for straws and packaging, which could also be commercialized in the near future. And in terms of fuels, we are again at a very research stage, because for chemicals and fuels to be possible, we also need the biomass at a very large scale. So even to tip in the chemicals and fuels, you need millions of liters to actually cause some significant movement. So we are on the path to make that happen, which will be integrated with our cultivation technology, where we can grow seaweed at a very, very large scale and drop the cost of seaweed biomass. So just wanted to show the kind of research that went in developing the products for plant health. So we have published extensively on how the product works. So we have global patents on the active ingredient. We have tested it in more than 25 countries. We are actually selling in more than 25 countries today, with some of the highest certifications that we can get in terms of organic certifications, and showing that the product works across crops, across geographies in a very consistent manner. And these are some examples of the countries we are we are present in, where we have developed data, and it works across crops, at least 10 to 15% yield increases. And world over, biostimulants are now perceived to be a tool to fight climate change, to fight abiotic stress, to improve yield and quality of crops. So this is a very interesting innovation that actually translated directly into improving crop yields for the farmers from C6. And we also had another product line which we have commercialized, which can improve immunity in crops against viral diseases, particularly chili leaf curl virus, tomato leaf curl virus. I think some of the students from pathology may relate to that. So this is a broad summary of the kind of research that we have done. More than that, we have also worked with a lot of multinational companies to actually take the product from lab to the final field in many of these countries. And where we are today, last year we clocked about $7 million revenue. This year we are close to $10 million in revenue, mainly from the agricultural segment. We have covered close to 8 million acres with our product, touching many farmers to help them improve their yields. We are present in more than 25 countries with 300 plus people globally, and more than 13 patent families covering all the technologies that we are developing, both in seaweed cultivation and seaweed processing. So, and again, lot of products in the pipeline in other divisions like food, biomaterials, and so I can say that we are one of the only companies to have completely focused on integrated seaweed supply chain, as well as developing and creating a completely new economy from seaweed, which was not exist before. So some key learnings from where we started to now, I think, especially because a lot of students and faculty may be interested in developing products to take it to market. I think a lot of it is at the intersection of value proposition, regulatory, freedom to operate. Can you actually make it at scale? So all these are considerations that go in when you actually design a product. So along with that, of course, you need to have a fantastic team who can support, who bring in complementary skill sets to contribute in different. I mean, it was not just possible to build this only with research and development. We had to actually invest in manufacturing. We had to invest in supply chain of seaweed, where we put in money for farms in Indonesia. We had to build an entire ocean farming team. We had to build an entire sales and business development team, a regulatory team. So a lot of skill sets actually came in to make this vertical happen, and translate from lab to a business. And one thing I would say is, we were not all agri graduates, or we were not even aware of seaweed when we started C6 in 2010. It was completely new for us, but we were willing to learn and adapt and explore what it meant for the entire vision of the company. Right? We wanted to create disruptive solutions for the planet. So that eagerness and that ability to learn fast will always keep you ahead of anybody else who's in the same field. I think that was really key for us. And collaborations. I think one of the key examples that I described you was when we collaborated with crop physiology department for almost 3 years. And then we also had a long-standing 10-year collaboration with pathology department here. And similarly, we worked with a lot of ICR institutes, because you cannot provide and design all solutions yourself. You have to work with people around you. You have to fit in and leverage on each other's experience. I think that is where domain experts come in. Like, for example, the university may be domain expert for agriculture. We may be a domain expert for seaweeds, but intersection is where you can actually create and develop completely new products and new solutions, I would say. And being able to pivot and adapt, I would say, initially when we started, we were solely focused on biofuels as a product. But as we realized that the scale required for that to happen is very, very large, and for us to, we may not be able to invest that much in the initial stages of the company, we actually pivoted to exploring what is the revenue possibilities for C6. Because for a company, you need to be able to stand on your own feet. You need to be able to, you need to be sustainable. So that I think being able to pivot and look at new opportunities with an open mind is really important. And lastly, I think, this field is a very long-term field. I would say biotechnology. You need to at least dedicate 5 to 10 years to see a product come out. So the first set of experiments that we had for developing biostimulants was in 2013. So for us, to actually launch the product was somewhere in 2016. So it took us at least 3 to 4 years to figure out how the product works, whether the product actually works in different crops. I'm just giving an example of agriculture. So even if you look at other verticals of agriculture, it may take you some time. So perseverance and being able to invest in developing your technology is really key. And lastly, the team that you really need to have an interdisciplinary team with different skill sets who can come together to make that happen. And funding, of course, that is really key. You need to be able to either generate revenue, or you need to be able to raise money to budget in for all your requirements as a company. So that is really key. So those were some of the key learnings that we had as a company. So yeah, I just kept it very brief. Didn't want to go into too much technical details, but I'll be happy to answer any questions later. Yeah, thank you.
Thank you very much. Oh, so that was, again, you can see this a transition from IIT Madras to Agriculture University, and having their company at the backyard of our university. And that's the journey, very envious. I'm very jealous of them. And that's fantastic. But they did something which is very important. Once again, this kind of thought which will come. And again, as you were saying, the 14 years ago, that picture that you had, we have some of those young kids who are sitting there at the back, who have some of these silly ideas. At the end, Panmalagi and the team, at the end, after Aman talks, sit down now. At the end, after he speaks, one of you or two of you can come here and talk for 2 minutes about whatever that you have thought about. All right? Think about it. Very good. I mean, listen to them, and they have, it's a whatever idea. Let's, this is the first time young undergraduate children are asking questions in a technical conference. Very good. I'm very, very impressed with that, and hope this particular kind of discussion that we have in the panel would actually take you far. And I would like to see you, and I will be alive to see you after 14 years, a bigger company than Shailaja's. Yeah, that has to. Okay. So let's go ahead. And again, as I said, Gopi is one of our darling blue-eyed boys of the department. And I remember that he used to ask me this question when he was an MSc student, or maybe when he was in final year. His hypothesis was cancerous cells that become, malignant are superior. They are fantastic, and they don't want to live in the stupid colony called human body or any body. And they were so superior, they die. They kill the organism. Unfortunately, they also die. Why don't you understand what happens in the cancerous mechanisms? I still do not have an answer to that. And those are the kind of tough questions these guys asked. And Gopi has his own company now. I'm so happy about it. And Gopi, you can tell us, how did you travel from? See, they both of them, Praveen and Shailaja, have come from somewhere else to agriculture. And Gopi here from agriculture, he went to another domain and did well. So the moral of the story is very, very simple. We'll come back to that at the end after we listen to Gopi.
No, I have to present, but I can talk.
Oh, yeah.
Morning, just sharing. No, you can hear now. Yeah, so like this, probably to 93, around 93, 94, we're all undergrad, fourth year or third year, fourth year. We used to attend some of these talks, and then we used to have a lot of fun. So morning I heard Dr. Vishwanath telling that Nobel Prize is the key thing for everyone aims at. As soon as you become scientist, or you want to do science, one thing you think is you are going to win Nobel Prize. That's the kind of the thought you'll have. So during the course of learning, you know that Nobel Prize is not so easy to get. So it's a lot of hardship, and then you need to probably you don't even know where you are heading. A billion, we might have a billion people. We have probably 3-4 Nobel Prize in India. So that is the way it works. But what I say is, I started as a kind of again, I was very curious, and then probably we used to go and then hide the books, and then so that seniors should not get. We all students used to go, and then our batchmates, and then learn molecular biology, because it used to be 1 or 2 books during that time. So we managed to learn whatever we wanted. And then obviously physiology was the best thing for us. I joined physiology as MSc crop physiology, and I worked with Professor Uday Kumar as a student. So that is one thing. And then for PhD, I was able to get into NCBS, in the sense I had a CSR fellowship, and then Professor Matthew graciously took me as a student. And then Professor Uday Kumar was my co-guide, and then I got the best opportunity from both the places. So the bug is, okay, you have to do something different. So that's what the bug always used to be there with us. So though I was a plant molecular biology student, at that time I had a hypothesis called RDRP, which RNA-dependent RNA polymerase has to be there in a cell, because otherwise the cell can be more economical if the RNA-dependent RNA polymerase should be there. It's a hypothesis which I thought. And then I wrote a hypothesis, and then that got into the, finally, after 3 years of struggle, RNA was discovered around 1998. That is the time we republished that in the current science. So that paper got me to the place where I had to go. So RNA mechanism was kind of discovering at the time, and then I went to do my postdoctoral studies. and then worked on the microRNA, and then I know it's the lab is something which is not everyone's cup of tea. So though you are hardworking, many of the times it depends on what is your environment, where you publish. All those things matters a lot to become a scientist. So while coming back, I got into company called Invitrogen. So the good thing is wherever I went, I started doing some of the new things. So that's a good thing in the many labs. So in Invitrogen, I was the first employee, and then I could set up certain things, work with them, and then do the stuff. So why I'm telling here is you can do multiple things as long as you have the life science skills and then the work on that. So that's where the molecular biology, what professor taught to us, and then how to be patient and then learn whatever is possible. That's the one thing which we all learned, and then started working on that. So I did a lot of genomics work I did as a technical person, and then also the business head in the company called Genotypic. So obviously genomics was exploding. Then we wanted to apply genomics into the multiple areas. So how do you do it? Sequencing was quite a bit working everywhere. And then I wanted to work on the translation research. That is the key thing which we were looking at. How do you apply the genomics or the transcriptomics into the translation research? We looked at the many technologies and also the problems. That's where the cancer, I came back to the cancer, because that's where I started asking certain questions, because we all know we have 38 trillion cells in a body, because there might be 100 cells or initially one cell starts to live, they want to live longer. So there is no wrong in cancer cell becoming, or they want to live longer. That is the basic fundamental rules of any DNA. If you look at it, a virus can go and then multiply as many number of times as possible. The same way cell has to multiply. So, but they are in a system called human body. So it's always controlling system. So it's a symbiotic relationship the cells have. They will survive and then they grow. As soon as they go out of the symbiotic things, the cells could be kind of get out and then they become a tumor. Tumor could be removed easily, and then or they could be killed, or they will come back and then metastasis will happen. These are the things which keep on happening. It's a cell in between the good cells and the bad cells. Always the fight is going on. So this is the thought we had, and then that's how the TheraQs kind of formed. What TheraQs is, a Qs is a signals for targeted therapy. So what each cell does is there is a lot of signals it keeps on giving, and then it circulates in the body. It could be a blood, it could be certain body fluids. So you have certain biomarkers, or it could be in and around this tumor. There's a thing called tumor and tumor microenvironment. We look at what type of changes are there in those environment, and then provide the right solution for the thing. So the idea here is, tomorrow, if any particular person, if somebody has a disease or something, especially in the tumor, if they go back and then if they look at the histopathology of that study, and then there might be one or two markers people are looking at, but they're not able to understand surrounding the tumor what type of changes have happened. So we have certain technologies we have adopted. There's a thing called digital spatial profiler. What it does is, on a section of the tissue, I can select the region in the tumor, and then in and around the tumor, and then take a call on what types of genes or the proteins are expressed in that, and then the pathways are changed. So the same thing, different technologies are coming in terms of the plants also. There's the technological stomics and other things. So we do work with many companies to help them to decide on the precision medicine therapies. And then again, with academic researchers we work, as well as the companies we work. We are around 15 people company now. We are self-reliant, but we are in the stage of growth stage now. How do we convert this knowledge, whatever we have gained from the tumor and tumor microenvironment, to a product or the analytics side? That's where presently we are working on. So that's the product side we are in the phase of developing. So if you look at the entrepreneurship, it's not so easy. So being a single founder, sometimes it's tough. Family, friends helped quite a bit. We didn't have the funding things. Obviously, even if you go to bank, as soon as you leave your job, your value comes down drastically, isn't it? So your salary is X, and then as soon as you leave your job, you're almost zero. Banks, friends, everybody looks at you as though you don't know, you don't have anything, kind of thing. So, but a lot of friends helped. Though if you know 3,000 people, there is only 3 or 4 people will be ready to help you. So this is the tough things. A lot of times you have to take call based on that. It's not an easy journey. Last 7 years have been fantastic. It's difficult. At the same time, it is really satisfying. We are ready to grow to the next level. We work with many hospitals at this point. We have many papers we have published for first time, collaborating with the TMC Kolkata, the St. John's. These are the places we work with them. Again, INSTEM and other places, Arjun and this, we work together. So the idea here is, one is, while during my PhD, I waited for 8 months to get a kit, one kit. Do you remember? Probably your project, only the money we got it. So in today's science, like we are almost equal to any country developed world. So the phase has 20 years, it has phenomenal changes have happened. A lot of technopreneurs have helped them to be where we are in at this point. I'll be happy to take more questions based on that. So, thanks.
Thank you, Gopi. And that is something which is very dear, because we have seen him grow from the department. So that is something which is very, very important. And well, as he says, I mean, somebody says that it is not a cakewalk. But have you ever walked on a cake? It's very difficult. So it is difficult for walking for sure. So before, you know what, I thought I would call you now to tell for 2 minutes, whatever that you plan. Come here quickly. And then when we talk to Aman about it, he will tell you whether it will be fundable or not. Isn't it? And then he will tell you where to sharpen your pencil. Come. You introduce yourself. I know you, but still, to the audience.
Good evening to everyone. We are third year students of BSc Agriculture from GKVK. Myself, Panbaregi, and he is Pratik.
I am Pratik Sagar, third year BSc Agri.
So with our mentor, Dr. Sheshai sir, we came up with the idea that is biofuel, which is produced from the bioalgae. So we thought of it because we are seeing more of carbon dioxide released from the industries. So we were like, how to tackle it? At the same time, we thought that we are importing more of fuels from other countries. So which costs more than $110 billion while importing that. So why can't we reduce that cost? So we came up with an idea. How can we convert this bioalgae into biofuel, which will also reduces the carbon content in the atmosphere?
No, you have to show it to the computer.
So these are the problems that we are facing. We are facing economic damages, weather problem, natural environment changes. And this is basically our concept. So we are using ladder system, and basically we are using algae. We will pump the algae solution to the top, and we have a ladder system. which will trickle down the water. So we are basically using the flue gas, which are emitted from the industries, which are very toxic, and we are using the carbon dioxide part. And other part, we can use anaerobic biodigester from the agriculture waste, and for enhancing our efficiency, we can use the culture of methanogens, sulfur forming bacteria, and other parts. So it will it will work faster. So this is the side profile of each of our ladder system. We are pumping the algal solution from this part solution, and we will take to the upper part, and there are flat nozzles, and we will trickle down the water. Basically what happens in classical system, that water goes down in the straight path, and it will not increase much of the surface area for the contact. But in this innovation, like we are using thrice the surface area. Like this surface area is there will be surface area from that part and that part, so it will give around thrice the surface area, and we will have the steep. And our second innovation is that we are using change wavelength, like red and blue wavelength, as it increases the photosynthesis. And it will be the source of it will be from LED, so we will not have any carbon dioxide or infrared heating that causes the loss in yield. And we are thinking of using chlorella vulgaris, but after research and development, we will try to enhance and change if it's needed.
So as you can see, with the help of the different enzymes, we can produce the bioethanol. And like you can see Saccharomyces cerevisiae, so it will be helpful in the production of bioethanol and as well biofuel. So so what are the main benefits of this technology? So since we are collecting the carbon dioxide from such industries, so we will reduce the carbon content in the atmosphere. At the same time, we can give carbon credit to the industries, and also we can give tax concessions for such industries, because since governments are having the policies that if we reduce the carbon content in this from the atmosphere while producing any products, so there may be subsidies or tax concessions will be giving. So when we collaborate with such industries, it will be like B2B economy. They are also benefiting. At the same time, we are also benefiting from this. At the same time, we can control the environment. It is in a controlled environment. So the contamination level will be very much less compared to in open environment. At the same time, these bio waste which will be produced from our technology, it will be given to the farmers. So what will happen? So when we give this bio waste to the farmers, it will in a form of biofertilizer. So it will increase the carbon content in the soil, and it will increase the yield of the production, either rice or wheat. So which will also tackle some of the food scarcity of our country. And the thing is, this is our work plan. So as you can see, first initially we will within 3 months we'll try to set up the initial setup, basic forms. Then after within a 1 years we'll try to produce the biofuel, and it will happen within 1.5 years with all the basic inputs like frame, cultures, and those things. And this is our budget distribution. As you can see, for one setup, it will take nearly 60,000, and for 60 setups, it may take 2 to 3 lakhs. So this is our financial analysis. As you can see, from one setup, monthly we are getting 3,400 liters of biomass, and while bioethanol which we get is 5,500 liters, and when you calculate it, we may get the revenue of 1,62,000. Then afterwards, the benefit to cost ratio will be 1.40, and the breakthrough point will be 1 year 8 months. With the total revenue will be 55 lakhs. So the byproducts will be produced will be manures, macromolecules, and biomaterials. This will be, of course, given to other industries like cement industries. So where we can include these type of ashes which we produce to them. At the same time, biofertilizer is very, very important in respect of food. Then the pharmaceutical industries also we can give. At the same time, our technology is a B2B, as I said, with the industry which we collaborate, and it will become as a circular economy. So it will also be scalability, where our initial cost will be very, very less, as we are taking the algae production is very less compared to other areas. And tax, as I said, tax congestion and eco-friendly, and those are the things which will happen in our technology. Thank you. Thank you for listening.
Very good. We will ask Aman about the funding for you. Good. Very good. We'll see. So, I mean, I just wanted to say this. I told this to the Dean agree that Dean postgraduate, if there was a competition among the postgraduate or undergraduate students about startup kind of things. And I said that whomever they had given a prize, let them come here and present. So it was just some kind of a audience. And for this particular panel discussion, it would have been nice, but I don't know whether it was communicated. But these kids I saw, I just said, why don't you? And extempore, all right. So, I mean, these are the things, you see the pleasure and pain of pain and pleasure, or pleasure and pain. What is the sequence? So it's the pain first and then the pleasure comes later, and whatever happens. That happens because, Gopi said very nicely, it all requires a lot of money. And here we listen to Aman, and he will tell us what kind of projects will Ankur Capital fund, and what is it that they actually want. It's simple. He gives you 1 rupee, he wants you to give him back 1 rupee plus something. And what is that something and how?
Sure. Thanks, Dr. Shesh sir, and thanks for putting me on the spot as well. Very unexpected curveball, but sorry. Yeah, before I kind of answer Dr. Sesha's questions as well, I just want to kind of give a quick introduction about Ankur Capital and what we do. And also wanted to kind of talk about, we talked about capital, but there are so many forms of capital at so many stages and so many levels it kind of comes in. So I wanted to talk about where we invest in, where should at least a startup look for, and how that capital kind of plays a role, etc. So, sorry, we have been in early stage startup investment space actually for the last decade. The idea that our fund was started is that we wanted to kind of see where technology is kind of coming and playing a role, and how it's kind of changing some of the underserved or underpenetrated markets in India, and how this technology can actually play a role in actually organizing these markets, enabling these markets, and then globally expand out of India to actually serve a much global need as well, right? So there was a broad idea that we kind of started with, and when we when we looked at what are the markets, what are the underserved markets that we saw technology will play a role in, we have we identified agri-tech, we have identified biotechnology, we have identified health, fintech, health, education, and a few of the areas, and few of the subsectors, some of the problem statements in those areas as well. So that's how the journey of Ankur Capital has unfolded, and since then we have invested in over 36 companies, and we have seen them kind of scale. We have seen them kind of struggle to scale as well, and some of them kind of shut down and move on to do something else as well, right? So the companies that scaled, they scaled well in India, they scaled globally. So and we are very happy to say that right now 5 of our companies are actually truly global companies out of India, working in multiple geographies, etc. So what we actually do in terms of our investments typically range in 2 types, which I would say first thing is that we don't typically invest in like a small scale or a company that would be like more operations heavy, or which would which would be like equal to a legacy company. So like we wouldn't probably invest in a restaurant chain, or we wouldn't probably invest in someone who's actually sitting by manufacturing unit, etc. That's not who we are, and I will explain the reasons for that as well down the line. But where we invest in is that we invest in digital technology led companies, someone who's actually using digital technologies to actually basically unlock new markets or serve existing markets. So this is your fintech, agri-tech, health tech, ed tech kind of companies. We have even invested in a sports tech company because they are doing amazing work on the ground, leveraging the new 4G infra and 5G infrastructure that's kind of coming into India, and how do you actually bring a lot of these grassroots sports to the global audience, etc. At the same time, we also invest in deep science led companies, and these are actually your biotech companies, your energy companies, or life sciences companies, where they're actually doing their R&D development. So we come in, let's say when they're at a TRL 5 or TRL 6 stage, we kind of invest in them because we like the team, we like the technology they are working in, and we truly believe that this technology will have a global implication. So we kind of work with them with our capital, but also work with them with whatever expertise we could offer to complete their technology development, go through their pilot trials process, and then eventually go towards commercialization of their products as well, right? So broadly, these are the two themes that we kind of invest in, because we realize that these are the two major opportunities that are emerging out of India. And we also felt that this is the place where our capital, whatever capital that we can bring in, we put in around $1 million to $2 million in the first investment. We just felt that this is where our capital would be the most impactful, that can deliver the maximum impact at the end of a certain time period that we look at as well. So if you look at our portfolio now, we have invested in, for example, an agri-tech companies which are in the B2B fruit marketplace, B2B seafood marketplace, truly global companies today. We have invested in food processing platforms. We have invested in inputs and output market linkage companies, etc. Even in fintech, we have invested in supply chain financing. We invested in an insurance company which develops parametric insurance products for agriculture or small farmers and multiple other kind of use cases. And on the deep science side, we have invested in a synthetic biology company which actually develops biostimulants from methane, and then actually converts them into your agri inputs or animal feed or human nutrition products at the end of the day. We've also invested in a zinc gel battery company which is actually developing a zinc gel battery which is used for stationary use cases, so replacing your diesel and DG sets or lead acid batteries, etc. While they might not replace lithium ion or high speed mobility use cases, but they're targeting a very different market. We also invested in companies in the health space. We have a company called Denome based out of Hyderabad. Again, researchers coming out of university, but they're actually using LAMP technology to actually develop point of care, RT-PCR replacement tests, right? So instead of actually going through the 24-hour or 48-hour period with RT-PCR, how can you use the LAMP technology to just do that test at the point of care itself? So they have significant implications in human or animal in healthcare in terms of figuring out communicable diseases, etc. And so we have done a fair bit of these over the last kind of 10 odd years. So that's just quick about where we invest in. I think the startups probably that you guys come across kind of fall between agri-tech and biotech for us. And we also realize that evaluating the deep science companies would require deep technical expertise. So we have a team of a couple of PhDs who are part of our team who actually spend a lot of time on the technical side of things. And I and a few others, we look at the business or the product side of things as well. But I think the topic I think we started out is just talking about capital and how what role a capital plays, and how do you actually, what are the parameters that you need to fit to actually get access to capital. So there are multiple, as I mentioned in the beginning, there are multiple levels of capital. You have your grant capital, which is basically given in a very early stage when you are in the idea stage to kind of get that idea to bit more maturity, maybe get a product ready, maybe actually prove a certain a point that you are trying to make, right? Then you have the venture capital where it comes in. Then you have the debt capital as well. The debt capital is when you have a scaled up product where you want to actually increase your rate of production, or you want to set up new manufacturing units, you want to buy new equipment, that's when typically kind of like debt capital comes in, which allows you to scale. And between these two is where venture capital kind of comes and plays a role. And the idea of venture capital is pretty simple. We go after the most ambitious and the most high risk investments that they're out in the market. So if you look at the history of venture capital, right, it started out from Stanford University. It's in 1968. There were a team of 8 researchers who are working under a Nobel laureate who wanted to come out and start a semiconductor company, because they were doing amazing work in semiconductor space, but they felt that like their professor is actually holding them back only on the research aspects. They wanted to create products. They wanted to create semiconductors which will go into our everyday appliances, your fridges, your televisions, your all of these like products. So, and they went out, then they went out and looked for someone who will give them the capital to kind of start this business. So as soon as I think earlier mentioned as well, right? As soon as you leave your job, your capital, your social capital kind of goes down. So the bank said no to them. Their friends and family kind of said no to them. Everyone thought this is insane. Why are you leaving a Nobel laureate to kind of start something of your own, right? So that's when they realized that they need a different set of capital out in the market, which is actually more adventurous, which can actually take the highest amount of risk, because this is a high risk idea at the end of the day, because you're coming out of a stable job, working under a Nobel laureate, to set up a factory to manufacture semiconductors, which may or may not at the end of the day find applications in different devices. So this is a story of Fairchild Semiconductors, which is the first case where venture capital come into play. So where a group of industrialists decided that, okay, hey, this is an interesting idea because this is where the future lies. And this is where the market is actually going to be in the next 10 to 15 year period time frame, where our appliances are going to shrink, our cars are going to become more smarter. A lot of these applications are coming up, which means there is a requirement for a semiconductor like this. So they came together to actually make the first known venture capital investment in this company. That's the name where it comes from, right? It's supposed to be adventure capital, but they removed that ad and it became venture capital actually. So that is the kind of companies that typically venture capitalists look for, right? So if there is a traditional company which has a track record of 30 years to reach a certain scale, we are looking for companies, the new companies who are coming in, who will actually reach there in the next 4 to 5 years time frame. So that's the benchmark that we evaluate companies against. Or if you are creating a new innovation that that hasn't existed before, we are looking for people who will actually come in and have the expertise, have the dedication to realize that innovation and then launch commercial products within, let's say, a 6 to 7 year kind of time frame. So that's where venture capital lies, and that is the benchmark that any startup that comes to us, we actually kind of evaluate them against. Are you doing something different from what your earlier people who have been in this space have done? Are you actually going to actually move that faster, execute that faster? Are you actually going to collaborate with the industry much faster, iterate and pivot much faster that you require? And most importantly, what have you figured out which is different? We are not we don't expect, as early stage venture capitalists, we don't expect them to prove what they have figured out, but it's about saying that what is that unique customer insight or unique industry insight or unique technological insight that you have, and what is the rate of belief in that insight that that the team is actually coming to us with. So these are the few parameters that we kind of look at when it comes to venture capital. Obviously, as I mentioned, there is grant capital and there is debt capital as well, which works alongside us essentially. And even in venture capital, there are multiple levels. We are an early stage venture capitalist, there is growth stage, and there is obviously late growth stage or private equity stage, depending on the where the startup is and what is the level of their requirement, right? So typically we come in at a very early stage point of view. So we have invested in companies which which just got created 3 months ago, and they haven't done anything so far, but we liked the founder, we liked what they have figured out in the market, what they understood about the market and what they wanted to achieve their vision. So we are just backing that. We also invested in companies which have been existing for 5 years, right? They have struggled for 5 years to figure out a scalable product, a new customer insight, something that can actually disrupt the existing market. So we have invested in them as well. So it comes in all shapes and sizes, I would say. But broadly, yeah, I mean, this is the most ambitious form of capital that comes in. And we are here, the important thing to understand is that we are underwriting a significant risk, because we are going after the most ambitious solutions. So we are the costliest capital that is available out there, because we take a part of your company, right? We take a share of your company, and if you are successful, that share is a lot, a lot of it amounts to a lot of money. So you have grant money, which is basically given, it's a non-dilutive capital, which means it is actually given to you without any return expectations. You have debt capital, which you get like 15 to 20% or maybe 10% like an interest rate, so which is also not that costly. But venture capital is actually the costliest form of capital out there. So, and the expectations also correspond to, right? So that's broadly that I wanted to kind of make my point. When you think about actually going towards a VC, and when you think about going towards like a venture capital, just few things to keep in mind. This is the costliest capital. We are looking for the most bold ideas, the most adventurous entrepreneurs who are actually coming in place. And we are going to judge across multiple things depending on the stage you are coming in. We're going to talk about the founders. We're going to talk about the idea, their vision. We want to kind of talk about what is that unique insights that you kind of figured out, and how this journey is going to kind of play out, right? So here we are essentially underwriting a lot of risks, while there aren't enough proof of concepts, but that's essentially what venture capital is, and that's what we kind of do, hopefully. So coming, I think, as Dr. Shesha sir has requested, just talking about taking the earlier presentation as an example. Again, I think boldly, it's a very interesting idea, actually trying to use industrial ways to kind of give back to the industry. This is a perfect example of a circular economy. And one thing I would say to the entrepreneurs, as well as they're actually building this out, I think in the presentation you have mentioned few of your assumptions around tax concessions, carbon credits, about the cost that it takes to set up the units, and what are the return expectations. So any assumption that anyone takes when developing an idea, the most important thing to do is that you just question the why. You just keep questioning it till you are 100% convinced that this will actually work. You actually go and talk to hundreds of customers, talk to industrialists, talk to anyone that you have to talk to, but just try to build conviction that this assumption, if you are making this is actually going to work. This is the way that it's going to work. So that's actually going to be very important, because these are the questions that you will get when you come and present to us as well, right? Because we want to know why this, why that, why this, why is this going to work, why is this not going to work, etc. So it, again, our confidence builds from your confidence. So just building that confidence plays a significant role here. And this is a software element, right? It's not some mathematics model that you are presenting, or something that you are actually showing. It's a very soft power element. And the other thing that you also kind of want to understand is what are the challenges that you will face when you are actually proving your proof of concept, or actually going towards scale up, right? Is it going to be a manufacturing challenge? Is it going to be a technology challenge where you will hit a certain barriers, etc.? Be open about it, be honest about it, on what are the challenges you are going to face, and how that's actually going to impact your growth, impact you, the size of the company that you will actually become. Let's say if there is only a certain level that your technology can actually grow towards, after which you will hit certain barriers, you realize that the market is just going to be that, accordingly your market, in the market size you are going after, will adjust. It may not be the entire industrial waste that you can use. It may not be the entire fume gas that's actually coming out, or your biofuel might not be for all sorts of equipment that are actually running, etc. So you just have to be very clear on the challenges that some of these things that you will face as well. So these are some of the clarity, or maybe whatever, even if you don't have clarity, if you're honest about it and clear about it, it helps us also understand where you are kind of coming from. So that will also help us a lot when we are actually coming towards an investment decision, etc. So I hope this is helpful, but yeah, that's actually what I could actually get in a very quick period of time. But yeah, thanks for the opportunity, Dr. Shesha, and happy to answer any questions.
Thank you. Thank you so much, Aman. That is really wonderful, because I mean, if you see what Praveen did, he went and interviewed some 60 villages, 8,000 farmers, and found out what really is the problem. He spent some time there. He understood their problem, and then they developed the solution. I think that's the clever way of doing. I think that's the kind of thing that the capitalist actually is looking for. I mean, unfortunately, the table that they made, this is the money that I will require, this is the time that I will take, was because the university suggested to them, make this slide this way only. So they put some number there. In 2 years, I will make 20 crore rupees. So you take a call. I think, isn't it? So I think that's a good suggestion that he made. I think that we'll remember even next year when we do the entrepreneurship competition, we will tell that to them. This is the way they should evaluate, not for how many years. I mean, it can take next year, you may make 10 million, or, in another 3 years, you may lose every damn thing, isn't it? Now, this is something which is interesting because we have successful people who have experienced the pain of going through, and then the pleasure of having developed this one. One thing which is common for all the three entrepreneurs that we have today on the stage is that they persevered. 14 years is a long time. It's a lifetime kind of an effort. 14 years is your imprisonment. No, what is that? Life imprisonment is 14 years. So that many years they have. No. That means to say what happens is that 14 years is a life-changing kind of thing. So I could see Bindu Madhava was discussing a lot with his friends. Do you have any questions to ask, my dear friend?
This was to Praveen, the first presenter. I was very curious and know about that, the kavach you wear while doing the spraying and other activity in the field. And I was working with the CGIR, and we always, proposed to wear that, the safety cover before you go to the field and do any activities of that. The one thing you told about that, we can wash it and reuse it, right? And suppose whatever the chemical you spray for that, all will be stuck to that, whatever the cloth they wear. Once you use the water to wash that get into the water bodies, right? That also chemical get into that stream and also again to the crop. But did you think about that? What is the solution for that? Thanks. Yeah, I think that's a great question. So what's happening is like the two things, right? One is wearing the physical barrier PPEs. One of the problem that is like, these are like a physical barrier, wearing under the sun for these people, like it's impossible, like, it causes so much discomfort, like in 5 minutes they will throw it away. So that's why it's very critical that like the fabric what you develop should be highly breathable, 100% cotton, so that like they can wear and like do farming for like 4 to 5 hours without having any discomfort. Regarding the chemicals, like they break down in such a way that like they can no longer cause any toxicity. So in fact, the people have the breakdown products, people have studied significant toxicity of those breakdown products as well, both on like human as well as the environment. That has been done. So that.
Ramon?
A couple of questions, one to Praveen, one to Shailaja. Can I?
Who do you want to ask first?
Praveen.
Okay.
Hi, Praveen. My name is Ram Mohan. I work for a seed industry at the moment. Excellent presentation.
Thank you.
Excellent. Thank you for sharing. First question is how input industry is aware of how much of your work is known to the input industry, because I was pretty ignorant. Okay. Yes, I don't work with the chemicals, but I'm also an input industry person. So direct question is, have you been in touch with any companies who are selling these chemicals, either directly or through any industry association?
Yeah. So regarding the awareness, yes, we need to bring that. Like, but this is like we launched a couple of weeks back, so we are pretty new in that way. So we have reaching to the people. Like that's the biggest thing. Like we started like as a pilot. We started working with like influencer in the villages. You have like these are like 100, 2,000 farmers networks they have, reaching through them to bring the awareness and all. And we started talking, we know that like, need to be commercialization. We need to do a lot more innovation than that what technology development we have done, because we are targeting the farmers whose purchase power is very low. And also it's a their thinking shift in their thinking, right? Because you can ask them to spend any money to protect crop, they don't mind, but protecting themselves, they think, twice. They don't even go to doctor to show them, right? So that's why it's a price point also, we put it in such a way that it should be affordable. And there are two things we are doing. One is we're trying to interact with the government bodies to see as a scheme, they can procure and distribute to the farmers, but which is might take much longer time and so on. And meanwhile, we have started interacting with some of the companies, both seed companies and the pesticide selling companies and all. Two things. One, they can procure and they can be like a distributors and sell them because they have direct connection to the rural areas. And also, temporarily, we can also do the CSR effort because there are a lot of CSR effort they do. So as a part of their CSR, they can purchase and distribute them. In fact, a couple of companies I already agreed to do that, close to 50,000 farmers, we are going to supply these sorts very soon through this con. So we are looking for, that's why any number is from 1 farmer to 1 lakh farmer, we are ready to partner with them to do that, either individuals or companies or any form should be great. So I'm happy to get connected and see which way we can work together to do, because our aim is to protect every single farmer in this country and the developing countries from this toxicity. Yeah.
Yeah, you have another question to him? Or?
No, just a bit of why I asked this question.
Okay.
Why I said it was there are a few responsible companies who try to. I completely agree with you to work with the mindset of the farmer. There are companies who are willing to put that extra money to provide these protection kits and along with their chemical, but the adoption is we fail to convert. Okay. So my point is, in fact, companies will be more than willing to partner with you to put this in their channel instead of really, because it's also an additional cost for the farmer. And if you can, if there is an opportunity, there are enough players really to work in this segment. We can take it up as a.
Yeah, I agree, because end of the day, if they don't adopt, doesn't matter how good technology dies, right? So we also had that consistently back of our mind, how it is going to be. But what we are observing is phenomenal, because many companies, they even Bayer and Monsanto and all, they also provide these PPE kits, right? But the compliance is very low because these are physical barrier ones. Okay, so if a little bit sunny, they can't wear this at all. 10-15 minutes, their skin get burned, they use it. So that's why the people started assuming that they don't wear it. But the reason they don't wear it is the discomfort. So that's why when we developed, in fact, the suit what I have shown, went a lot of R&D in the design of that and the breathability of the comfortness of that. Even we came up with a one single now size fit, so that a person from 5 feet to 6.5 feet, 6.2 feet can wear comfortably, and while farming they jump and all, right, so that comfort also should be there. We took all of them together. Now at least close to 100 farmers are using them, almost more than 3 weeks now. This is a season they are spraying, and we see a huge compliance because they also see instant results. Because every time they used to spray, in the night, they used to suffer, and with the pain they used to sleep. Now they don't see that at all. So it's the word of mouth also started increasing the people. So we strongly believe, if you ask me 3 months back, complaints, I would have still think, okay, that is just our biggest hurdle. But now I'm more confident to say that, if you ask me 6 months down the line, our complaints are going to be a lot more than what we thought about that.
As a chief of the IN, would you want to, if Praveen asks with you, would you associate with them?
Yeah, we would like to try it in house for our own in our own farm.
There you go. Yeah, sure.
Thank you. Because why not?
Yeah, I think Praveen, would you also be? Would you want to tell that the government of India, the agriculture minister, wants to release your product?
Yeah, yeah. Our science and technology minister, he wanted to
Science
launch, but Now we supposed to launch last week of the October, September, but the Kashmir elections was announced. So 1 month they postponed. That's why officially we didn't launch into the market. That's why we didn't see the news much yet. But like we are doing all the beta tests. Yeah.
No, he has one more question to Shailaja.
Hi, Shailaja. Again, great story. But I was aware of your work. Perhaps they were in Monsanto around 2011, 2012. I don't remember whether it was you, but to see the growth from then and to till now, amazing. Couple of quick questions. Okay, regarding this immunity products which you have, the plant immunity products, did you face any regulatory challenges?
No, so yet. So because we are trying to position them as plant health boosters, not as, because they don't directly kill the pathogen. So they don't fall under the regulatory profile of central insecticide boards. So we are trying to position them as a stimulants which can improve biotic stress. Yeah.
And was the Indonesian sea farm a strategic decision or why?
Why
India-
Indonesia?
India
Yeah.
would have
So for us as a company, when we wanted to grow our business, we also wanted diversification. So Indonesia is one of the largest producers of seaweed, cultivated seaweed. So India has like 0.1% of the global seaweed production probably. So we are not, and our country doesn't have the legislation to promote seaweed cultivation on the sea yet. Government is very interested to promote seaweed cultivation. In fact, every Independence Day, the Prime Minister talks about seaweed, but the real on-ground implementation is not yet there. So, but in Indonesia, they have a decentralized way of accessing sea surface. We actually leased a 1 square kilometer area on the sea. And we are able to like cultivate and take whatever machinery and concepts that we are designing in India to Indonesia for testing. I really feel that when that is a success, Indian government will really take a notice of it and implement legislations in India, so that as a industry we can also move forward in India. Yeah.
Is there any reason why Indian government is not thinking about allowing the sea surface cultivation?
It is not that, I mean, as a country, we are very land focused because we have a lot of land, lot of other problems to deal with in land. So as a blue economy policy is shaping up, I think the focus is coming in, but on ground implementation is not yet, is slow. I would say it is slow. So in fact, they allocated about 600 crores for seaweed cultivation programs in it. But the way it is being implemented, how we want to get there in the next 5 years, all that is very unclear. But all across the globe, there is a lot of focus on seaweed because it's a sustainable feedstock which takes carbon dioxide from air. You don't have to give any inputs of fertilizers. It's like, unlike traditional farming, you don't have any inputs. It just grows on the sea. But the only thing is you need to be able to keep the seaweed on the surface.
Exactly.
So,
and when we were working closely with the physical research laboratories at Ahmedabad, they were more interested in what is called the primary productivity. The concept is that the sea surface is 3/4 the globe, and on land is only 1/4 of it, right? So they said that 3/4 of the global productivity can come actually from the ocean, but the truth is not at all that. The growth of the plankton is only on the shore, where the riverine discharge happens, because the deep sea is significantly devoid or deficient in iron. And the growth rate of planktons is extremely poor there. So that is something which perhaps would be one of the reasons. I do not know if Indonesia has a lot of rivers which will flow, bring in a lot of nutrition to the surface, and therefore the productivity is high. I do not know how productive.
actually, they have a lot of islands. They have 17,000 islands.
Yeah,
If
there
you
you
did
go.
not, so it is a very interesting landscape and it is very well protected. So it has, I mean, that country has taken seaweed cultivation very proactively. And
Yeah.
even at the highest levels in the ministry, they are very focused on making this a national priority, actually.
Yeah. And the Indian coast is extremely smooth and all that. And therefore, would your company also want to sort of improve the seaweed itself to grow in, let's say, iron deficient conditions? are nitrogen deficient. Like we try to do that in the land as well.
We are having some collaborations where we are trying to better the seaweed species, but that is a very long-term approach. And since these are vegetatively grown, whatever we do, even if we do it by ourselves, once it grows out into the ocean, we cannot control who uses it. So we are trying to do this in a very public-private partnership kind of a model with some universities. So we're trying to create that correlation so that more and more people actually can notice that this is a very interesting and useful raw material.
Absolutely. Guru.
The question is to Mr. Praveen. A very noble cause of actually protecting the farmers from pesticides. But the same pesticides which are sprayed on the crop finally also end up as a pesticide residues in the end products. Would your technology work on the on reducing this residues in the end products? In that case, a number of industries probably would be willing to really take it up, invest, because most of them actually would like to showcase that their produce actually is pesticide free.
I agree. So we did develop a completely different technology to deactivate the pesticides in the food, but like it would work only on the surface. If you take vegetables or fruits, now we have a solution like if you wash with this one, it will completely deactivate. But the problem is more often like your surface pesticides are very little amount, unless like a grapes and all you have highest amount. But like the problem is like it's already seeped inside the vegetables and all. So those ones like we don't have any technology now. Like the question is we can use this technology, but like the kind of regulatory body it has to go through use that, because then you're consuming this solution also, right? So if you're using for the deeper one, so bringing that to the market is like a very different regulatory path one need to go through.
Certainly appreciate that. I was actually looking more of something which is already pulverized, let's say like atta, right? I mean, it is no more inside. It is, I mean, the whole thing actually should be exposed. Your surface area should be already available for interaction. I mean, barring the regulatory hurdles that you have to go through for the food use, would those be some aspects
that could be used? We have already solutions created that. Yeah.
Ravi? No, I think Ravi, one second. Dr. Vadan Pal wants to ask a question.
My just one suggestion is to the Praveen, basically. First of all, I congratulate you to develop a very good material.
Thank you.
And that to a very noble cause. Regarding the cost of this 3,000 rupees, we know that it is not very high. But once you look into the perspective of the farmers, you said very clearly that they are very measured for their own safety.
Yeah.
So my idea is there because farmers are buying the large quantity of the pesticides from the dealers, maybe the 10,000, 20,000 like that. So if along with that one, to promote the sale of this item, if you give some promotion to, with the dealers that if you buy the 10,000 rupees, it can be given free of cost. Then they can be having the more interest to purchase it. And that can be useful.
No, absolutely. There's a reason we started engaging with the pesticide companies, so that they can purchase and they can distribute, and they can in fact they can say increase their sales as well by giving this and protect the farmers as well. So we are exploring every mechanism that is possible to see what is the maximum number of the farmers we can reach them. And also we're also thinking another like a community farming type. For example, it's not like that every single day every farmer spray, right? So you can have one suit, maybe in that locality 10 farmers together, they can have it. Whenever they spray, that person can use it because it's highly washable, reusable. So absolutely, that's one way to bring down for each farmers individually. If they want to use it, we can bring
taking care of the because semi-arid regions like during the season of the rice and our North Indian conditions, it is also used in the summertime for the sugarcane. So if it is not having any problem with the weather, that is it can protect them from the humidity and the temperature, then it can be used very frequently.
Yeah, absolutely. In fact, the quality of the fabric keeping on all those parameters.
Okay, thank you. Another query is Selja. Do you have any information related to the application of your product for the abiotic stress tolerance, like the drought stress or the salinity?
Yes, we do generate some data there. on drought stress. Any reason you had asked?
In the research experiments or in the farmers field?
No, we've only done research experiments. We have not actively pursued that in the farmers field. We plan to do that in the next year.
How was the response of that one in the application?
It's very encouraging. Like we have done some trials even with IHR where we have reduced the water usage.
And another idea is that 15% actually is not very acceptable. So
sorry,
10% or 15% that advantage by the application. In that one is not very encouraging one. So you should have the product in such a way that it should go beyond 20%. Then it can be more.
In some crops, we do see more than 20%, but globally, I think 10 to 15% is acceptable
Yeah,
for
averagely
products.
10-15% is okay.
Yeah.
But if you look into that 10-15, it looks very low. So always appealing if it is more than 20%. Okay, thank you.
It's like juggling. Somebody was juggling with 3 balls. Why not 4? Why not 10? Why not 100? So that's how it is. But I think that's a point. I mean, more the merrier for sure, sir. I mean, whatever they are doing has been showing some good responses for all sorts of abiotic stresses. And it will be interesting. The reason why Shailaja is here and their company is very appropriate is also because we are trying to talk about the growth stimulants which are elicitors. Either you stimulate or elicit the response. I want this kind of response. That's how it should give. And these seaweeds seem to have all that, those hormones and whatever small molecules. Has anybody done any sequencing of that and found out a metabolome sequence of whatever that contains?
Of the seaweed? No, I don't think anybody has done metabolomics.
There you go. So, I mean, if that can be done.
There were some institutes which have done some parts of the seaweed metabolomics analysis, mainly like flavonoids and other things, but not the full seaweed. Yeah.
Okay.
I think what we have done is we've identified specific molecular ingredients which are directly contributing to activity. I think probably the first one of its kind coming out from India, going global.
Correct. That I completely agree with that. That's interesting and well done. Congratulations on that. And good that they are at our backyard, Rame, you have a question.
Yeah, a couple of small questions to Dr. Shailaja. The first question is like anything that we basically take from the ocean will have heavy metal contamination. So are you taking care of that? Otherwise, it's basically contaminating the food.
Yeah, so we look at the global regulations for such products and we meet all the required limits when we process our products, when we process the CV to final product. So we have multiple product ranges in biostimulants and biodefense. So we were able to adhere to that limits, which are within the safety limits prescribed by the government authorities.
So basically you remove some of those heavy metals when you process
for specific verticals like food, we have processing technologies that can remove the heavy metals also.
Even something which is going onto the plant as well. So if you have heavy metal, it gets into the food chain, right? Right, sir?
Yeah, that's exactly what I'm trying to say, that we have processing technologies which can address and remove the heavy metals so that the final product which goes to the customer is within the limits specified by the government.
Nice. Another business question. So there are big players already with seaweed extract like IFCO. You might know they are selling and going in a bigger scale in the name of Sagarika or something. So how do you manage to get your product to compete with them, for the...
Yeah, we are in the open market. We compete with a lot of players.
So how do we compete with bigger people?
Sorry?
How do we compete with the bigger people? So IFCO is known by everybody, and then generally, for people like us as a startup, so we want to get our similar product into the market. So I mean, other can also answer. I'm sorry.
Yeah, sure. I think it's an important question. Let's say there are already established players. We entered this space very late, actually. And how did we actually manage to sell, and how are we actually continuing to sell? Because market is very brutal. It doesn't allow substandard products to continue to exist, right? So you need to develop a really high quality product that can stand the test of time. I think that is where our understanding of our active ingredient, our ability to produce the active ingredient consistently batch after batch, really convince some of the large global multinational companies to come in and partner with us. Like we partner with FMC. We work with some of the leading companies in the country, large fertilizer companies, and companies. I think some are in the public domain already, like Yara, We work with FMC, we work with Zuari, we work with PI Industries. So I think the quality of the products and the fact that they work consistently and the fact that we have invested so much research and development into establishing the efficacy on the field will speak for itself in the market. And I mean, you have to compete on cost at some point. That is where our innovation in terms of supply chain linkage back to our own farms and our own method of processing also come into picture. So, I mean, all of this will speak over time. Yeah. So that is why we are continuing to grow. I mean, yeah.
Thank you.
You have a question.
Again, the question is to Shailaja. What is the consistency of your product from different purposes? Whether you are using same product across various purposes, or you have a range of products for various purposes?
Sorry, can you clarify what various purposes?
For water stress, you are saying you have the product.
No,
you are testing.
we are not selling any products for water stress right now. We are only selling products for improving nutrient use efficiency and improving growth and yield. In the biostimulant segment, that is a one active ingredient. We have another product line in the immunity segment, which is completely different from what we are selling in biostimulants.
Okay.
So we have two active ingredient platforms. Yeah.
Across the crops, same product you are selling.
There will be formulation differences slightly, but can I get the context of the question, please?
Because I am interested to know for fruits, vegetables, or cereals, are you suggesting the same product or you have a different products?
The product works across different classes. So we have tested it across different crops and it works uniformly. So you have to tweak the dosage and the application timing, but it is uniform because it is universal response because we are seeing similar categories of genes getting triggered across different crops. So we do gene expression analysis on different crops also, different model systems.
Okay. Second one is, now you are sourcing the material from Indonesia. There are questions that why not to use the material from India also. Is it going to make the difference, the product, if you extract from different places?
No, as long as the species is same and the active ingredient chemical structure is the same, it should not matter. Yeah.
Thank you.
To add Prakash's question, in the morning, Vishwa was answering some question asked, why are you working with Arabidopsis, why not rice? So when you have this ABA sprayed on rice or Arabidopsis, that the physiological response would be more or less exactly the same. So same here, I guess. And I think when you promote growth and yield under well-watered condition, whatever physiological changes that it would have, I think we'll have it even on the drought condition. I think if your product increases photosynthesis, why should it bother whether there is irrigated or stressed? So I think same product could do, but I think that was a good answer. What we need to perhaps do is see, they have a compound. What perhaps we should do, from the agricultural sciences, how is it that we can, I remember this when Shivu and I were doing PhD, we had developed some very, very ordinary device for developing CO2 response curves long ago. And somebody came there, we were young people, we were looking for somebody to appreciate. And instead of that, somebody came and said, "Isme kuch gadbad hai. Isko theek nahi hai." And before I told you what it is, gadbad kaise pata chal gaya aapko? So this was a question, you remember, we asked that question, Shivu. So now, what I am saying is, Prakash, I think what we should do as a collaboration is, if they have a stimulant, we should find out what it stimulates, how it stimulates, and where they can apply that, so that we have the win-win situation for both of us and our farmers getting benefited. That's my opinion. Anyone else? Purushottam.
My question is to Praveen. Do you have anything herbicides also? Or weedicides?
Herbicides, we don't have anything right now. Everything is like for the insecticides. But the second generation will bring for the herbicides, because there's a completely different chemistry what we need.
And I have one more question to Shailaja. Among the byproducts of your seaweed, have you found any major nutrients as a byproduct?
Byproduct meaning?
From seaweeds.
So seaweed is rich in potassium, definitely, so that we are able to harvest and include it in the formulation that we are developing. So we don't generate any waste.
No. Have you, do you have plans of developing potassic fertilizers using seaweeds?
So we've actually introduced a category called potash from rhodophyta in fertilizer control, where we use that to supply some potassium, not all of it, all of the plant requirements. If you want to supply all of plants potassium, that may not be commercially viable, extracting it from natural sources like seaweed.
Good. You have a question there? Can somebody take the mic there to him, please?
Sir, this is not a question. It is a sort of clarification from Mr. Praveen. So you developed a fabric which protects farmer from pesticides and its effects. Okay, have you got any tax exemptions in this on your product? Tax exemptions?
No, not yet. We haven't reached them also. We
Now,
didn't
why I'm
explore
telling you is
yet,
that,
yeah.
so many services in medical field, they have got even services and also goods. They have got exemptions. They have got by way of notifications. Okay, your product, it is protecting farmer, exclusively farmer, okay, and their health. So you are like-minded companies, you can approach the GST Council and sort for the exemptions also, both under GST and also the income tax. That is possible because many areas, so many these sort of activities, they got the notification done and they got the exemptions. Thereby, the cost of the production and cost of the availability for the farmer, it will reduce. That is one thing.
Oh, thank you so much. I think that's a great suggestion. We should definitely.
Yeah,
explore
I
that,
want to
yeah.
introduce him.
He is
I would
Mr.
love to.
Munindra from, he's an alumnus of our department, and now he's a joint director of commercial tax.
Oh, fantastic.
So, hey, Guru, give them exemption.
It is not in my hand.
Take it into your hands.
No, they
please.
have to approach the GST Council.
Yeah, do that.
They get it done, the notification from that.
Fantastic.
So whatever these related fields, no, even Gopi also, certain activities in respect of cancer, we are into that molecular biology aspect. Such activities all put together, even in medical field also, in a higher forum, you can approach the GST Council and get it done. See, such activities, many of the medical services and the products, they are exempted from GST. Okay, even Madam Shailaja also, because it is a bioproduct, right? So, and see, whatever it is originated through that sort of activities, you as a companies put together, you have to approach the government. Okay, you can get the benefits. And one more, the clarification I seek from venture capital. See, most of the people, they are going to start up as a beginners, okay, companies. And the capital is most important thing for any activity to start, okay. And the government schemes, there are many government schemes, like MSME schemes and all other things. And in both central and state governments, they are having many low rate interest schemes and all other things. And you are Ankur Ventures, no? You are Ankur Ventures.
Ankur Capital, yes.
What is the rate of interest that you are going to give them?
No, sir, we don't charge interest. We basically invest in the company. We take a share in the company.
Okay.
And as the company grows over years, like our investment value also grows.
Okay, okay.
Yeah.
You are a shareholder in that company.
Yes, we actually take share. We work very closely with the company on bunch of things actually.
Okay.
Yeah.
Nowadays, many shell companies are going to come into the picture. So how you are going to handle such situations?
So which companies?
Shell companies.
Shell companies.
Yeah, they'll.
No, we do proper due diligence. Like we have a proper legal, financial due diligence before we invest, and it's actually 2 to 3 months process with lawyers, financial accountants, everything, just to make sure there are no risks to our investment. Because we also raise money from our own investors. Like we have investors.
Yeah, it is an investment only because you get the money from somewhere and
Yes.
you have to give interest for them also. Like.
Yes, so we have a fiduciary responsibility to our investors as well. So we take that diligence and everything very seriously as well. So
Okay. My request is that see many the environmental friendly the industries which they are coming up, no, they have to be get benefited by way of the rate of interest and also the your efforts is very much required to approach the government and get the SOPs or whatever the benefits from the governments. And your product is patented.
Yes, we have like a granted patents.
Your
Yeah, these are like multiple granted patents we have.
Okay.
Yeah.
No, if you have got patented, no, so you can expand your thing based on the demand and supply.
Yeah.
Demand and supply. And when your requirement is going to be increased, when the cost is going to be low. Okay. And your CSR funds of your fertilizer companies, they have to take care of such activities.
That's right.
CSR funds will be there. In addition to that, the tax exemptions are very important to bring down the cost of the product. Okay.
We'll certainly explain.
Yeah, I mean,
Thank you. Thanks for-
in the interest of time, we will discuss that in the evening again. And there are some more questions. I'll ask that young person to ask a question now. Introduce yourself, Bata.
Good morning, everyone. I'm Ritvik. I'm studying in third year BSc right now. Ma'am, my is to Saleja ma'am. My questions are mostly probed on the production aspects of your business more than the other things. So I'm majorly concerned about the prevailing ocean conditions regarding to the ozone layer depletion and the changes in weather. How effective are you able to produce the like in the case of mass production of these vegetative growths? How effective?
You mean seaweeds?
Yes, ma'am.
Yeah, they are already grown at a large scale, more than 2 million ton scale in the Indonesia.
Ma'am, like there'll be some prevailing conditions. Like, do they grow even well in that result?
What do you mean prevailing conditions? They're growing right now.
Like they could be like mishaps, like in case of tsunamis or increase in wave sizes.
That is nobody's control, right? In normal conditions, they grow very well. There's no issues.
Ma'am, another, I am not sure how good is it, but they are high in carbon content, right? Yeah.
What do you mean high?
Like,
what is your question? Continue.
like, are there any threats to attack from sea creatures?
You mean something else eats the seaweeds? Yeah, there are fish which eat seaweed. So we pay fish tax when we grow seaweeds.
Okay.
Maybe like some 5-10% fish tax. Yeah.
And the last one is regarding how beneficial is it to medium and low marginal farmers? Like your thing is majorly it focuses on bio.
Sorry, your question is on what farmers, which farmers, land farmers or sea farmers?
Land farmers. No?
Okay. Yeah, we do give a cost benefit of 1:3. I mean,
okay.
At least to farmers. So that's taken care of.
Ritvik, if somebody is growing something in the ocean, what has got the land farmer to do with it?
No, sir. Products. Products.
I mean, after some time, there will be ocean also, marginal ocean space, possibly.
No, we always give benefit to farmers. These solutions are designed so that farmer gets better yield and better soil health. So it is not, it is not that the farmer applies and not sees benefit. Then there won't be repeat purchases from farmers, right? So
how beneficial is it compared to like the local products, non-organic products?
Non-organic products meaning? There are many products in the market. So you have to define what product. It is competitive with respect to any other product which is in the market. Yeah.
Ritvik, let me frame the question for you. Yes. Is that you wanted to find out when compared to chemicals, how the seaweed helps?
Yes.
Okay, ask again.
Frame the question.
Compared to chemicals, meaning plant growth regulators?
Yes.
See, plant growth regulators are used when very specific like you want to increase specifically some grape size, you use gibberellic acid, or specifically you want to increase some rooting, you use auxins, right? So they are used for very, very specific induction of certain phenotypic characteristics. Whereas what we developed and designed, they work more on the plant, holistic plant nutrition processes. So we see improvement in root phenotype, which is very similar to PGRs, but it is doing through elicitation within the plant. It is not directly hormones. So it should be comparable. I hope that answered your question.
Next question is to sir.
Oh, very good. Go ahead. Who to whom? Gopi, very good.
Sir, I was very fascinated by your idea. Since we two are a new upcoming startup company, I would like to have a thought of what is your plan of work and your way of thinking. How do you proceed? Through the 7 years of time, how have we proceeded? Majorly, I want to know what are your future plans with this technology.
Okay. See, our main aim is to take it to the patient level. So that is the main goal. That's where the scalable model, if you look at it. All these days, we are working with the researchers. From the learnings, that's where you develop certain tools and solutions, then go to the patient level. So that's where the regulatory things comes into the picture. As soon as you hit the human and then the patient, you need to have certain sensitivity assays, and then whether it is the effectiveness and all those things comes into the picture. So the regulatory is the next step. But again, we are developing the products at this point. We are not looking at too many of those things. Once we have the analytics ready, then we can.
May I know the exact vision of your final products, like 10 years down the line, your final product?
So our aim is to look at, okay, if someone has like someone goes with a cancer, let's look at this way, whether that could be treatable or how easily treatable is it. 10%. Now the chances of success is very low. That's where the immunotherapy comes into the picture, and some of the targeted therapy comes into the picture. In fact, I didn't tell you, Professor Uday Kumar inaugurated our company. So I am always indebted to do something in the direction. So that is one thing which we look at it. How, at least if we can save some people the pain of uncertainty in the drug treatment, that's the most successful way I can look at it.
And we lost our Professor Uday Kumar for that dreaded damn disease. Okay, before we ask Guma, before you ask a question, can I find out if there are any questions online, please?
Sir, I have one more question.
What's that? Important will come. Of course, everything is important. Who? Hey, wait a minute. I mean, you cannot take away the entire thing. We'll give you another chance, and others are also asking. Sit down. We have time, Ritvik. Very good. But I'm glad that you guys are asking questions. But is there anybody in the online, please? Online ಅಲ್ಲಿ ಕಾಣಿಸಲ್ವಾ ಅಲ್ಲಿ ಯಾರು ಇದ್ದಾರೆ ಅಂತ ಅದು ನಿಮಗೆ ಬರುತ್ತಲ್ಲ. There are some people still online. Sajivan, I can see Saji. Can you unmute yourself and say something?
Myself? Shashi, are you asking me?
Why not? Yes, we are asking you. Saji, you have any comments, any question, any suggestion? What are you up to? What are the things behind some pipelines and all of that? Now. Okay. When the online people are preparing, there are a lot of things on typed, I say, on chat box. Oh, okay. All right. So when they're preparing shoe.
Yeah. My first question is to Praveen. Here, not online.
Yeah.
Regarding your the PPE kit which the companies sell, along with the pesticides. So I think that itself costs around 1,000 rupees or something like that. So your product, since it gives protection against the pesticides, why don't you sell it through or make an agreement with these farmers producer organizations? FPOs are there. FPOs usually, each FPO has got more than 500, 600 farmers as members, and they have capital given by even government as well as their own share capital. So they can have a MOU with you and then take it, and then they can give it to their own members. So that one possibility of expanding your scope. And if it is launched, we also want to, as ICR institutes, we also want to purchase those things and give it to our own laborers who use pesticides in the field.
Thank you. In fact, that's why every possible way we wanted to get access to, I need to spend more time at Agriculture University, get connected to these organizations, because give that connected is very important. So I would love to get access to those people, so where we can go and demonstrate and give them more awareness and bring them. And ICAR institutes are absolutely, in fact, IHR from Bangalore, they've already procured some. So Balakrishna, so he already started a pilot study. He's using phenomenal background, the feedback they are giving to us. So we'll be coming to and spending some time there in IHR very soon. Yeah.
Thank you. Yeah. And for Shailaja madam, with respect to, see, when you started thinking about the seaweed products, first question comes to mind is which particular species or which particular seaweed I have to use based on their effect in published literature. Because I know that there are many species which are being used and which have been commercially used for extracting different chemicals. But thing is, which is more effective, which one I have to start, so that will be market accepted, and that will give better results. How did you decide?
So very interesting question. So when we zeroed in on the species, our main focus was on trying to work with species that can be grown, because we were trying to build or look for a species or a biomass source that can be scaled up infinitely. I mean, given all the resources. So our approach was always that how can we use a resource that can be sustainably grown? And in that bill, only a subset of seaweed species actually fit in, which was all the right seaweeds which are being cultivated in Indonesia. And thankfully, one of the species was actually growing in India, in Mandapam, because of some previous efforts by some research institutes like CSMCRI and other people and CMFRA. So we use that raw material as an input to do all our R&D, but we do all our R&D and product development, and we saw some very differentiated results compared to the market standard products. And that is when we realized that we have something unique, and nobody else has this kind of an active ingredient ever. Even we scouted literature and we did not find, and that is also one of the reasons why we got a patent on the formulation, on the active ingredient formulation. So, I mean, many things happened simultaneously because we were at the right time, or we had the access to the right biomass resource in between 2010 and 15. Yeah, that was our thinking and approach. Because before we came in, seaweed-based biostimulants have been in the industry for almost 30 years, I would say. Most of the brown seaweeds based stimulants are already available globally. I mean, not just in India. But they're all from a widely harvested seaweed, and you cannot really say that the raw material that I'm harvesting today is the same as what I'm harvesting tomorrow, because or next year. And there is also a limitation on how much you can harvest globally, because each country has set some limitations on, you can only harvest, let's say 10 or 15% of the seaweed bed. You can't harvest more than that. So for us, when we develop products, we want to target products that can scale up infinitely. I mean, provided the resource constraint and all of that. So in that, only a few species fit the bill. We started with the species which we had in our own backyard. And we figured out that there was very little innovation done with that seaweed. So we found that there was a unique space where we could play, and we brought in our own biotechnology experience to process that seaweed and discover this new active ingredient. That's how it all happened. Yeah.
Is it based on only the biomass production or based on the active ingredient of the weed species? Because for the industrial aspect, when you look at it, it should be highly productive, biomass should be very high. Otherwise, if the growth rate is very, very less for the seaweed,
Yeah,
you
it is
cannot
one
use
of
it
the
commercially.
it is one of the fastest growing species. It has higher productivity than any land-based plant. Like we compare it with sugarcane is like about 70 to 80 tons per hectare. You can actually get 100 tons per hectare with the seaweed dry biomass. One of the fastest growing species. So it was very interesting for us. I mean, when we started this journey, yeah. We felt that this could be something very exciting and it could solve some of the major problems if channeled properly.
People who are giving the mics, please listen to me because there should be no crosstalks, Mr. Rame.
My question is to Amanji. In your experience, what is the success rate that you have seen for the project that you have funded? And if you don't mind, what kind of products have been so far successful?
I mean, just looking at our experience, like kind of so far, if you look at a typical fund and over a life of, let's say, 10 years, we typically, if you invest in 10 companies, let's say, we see what we call 2 of them as winners. And 2, 3 more of them will actually what we call a medium, performance. And we would actually see 5 companies where, they might not give us returns as an investor. They might become medium-sized companies, they might be doing well, but it will not increase our investment value. So and I think 2 to 3 of them would actually kind of shut down etc. as well. So it's a mixed bag across these things. Regarding what has worked, I think we have seen success in a lot of companies which are doing in the B2B supply chain market linkages, in financing. We have seen in food processing, in biotechnology, in multiple in area sectors, right? So success for a startup essentially comes down to actually solving a real problem, having a good value proposition, timing the market really well, because you don't want to be ahead of the market or later into the market when there are a lot of people already existing. So you want to be at that very right time, essentially when the value proposition will play out, and then executing really well to actually become a very successful startup. So essentially these 3-4 parameters kind of come into play really well. Thank you.
Yeah, my question is to Saileja. Are there any restrictions for the harvesting of these seaweeds? The government has put any restrictions?
In India?
Yeah.
In India, it is grown by people, so there are no restrictions.
So you can harvest any amount of seaweed?
Because you can grow it.
Okay.
It's like any plant, like when you grow rice or wheat, are there any restrictions?
But harvesting so much of seaweed, does it really affect the diversity in the ocean diversity?
That is a question which is yet to be answered. So even in our pilot farm where we are laying out 100 hectares, the government of Indonesia is actually doing a biodiversity analysis before we started, and with due time course. We are not seeing any impact so far. It's been 6 months, so we'll have to wait for that because it will have to be done by third party. We cannot do that analysis ourselves, so we have to rely on independent reports that look at the biodiversity. But looking at the history of seaweed cultivation in Indonesia, it has been growing at that scale for the last 30-40 years. There have been no perceivable damage to the biodiversity. So I can say it is more contributing towards circular economy by harvesting all the nutrients in the sea. So I mean, the time will tell us. Yeah. Whether, I mean, there'll always be a tipping point, whether it is at that 100 hectares level or whether it is at 1,000 hectare level or 100,000 hectare level. We don't know the answer. Until we do it, we don't know. Like even when we do large scale agriculture on the land, there will be biodiversity issues, right? I mean,
maybe in the long run, maybe we may find the answer.
Yeah.
So,
But we are willing and we are open to explore that. Yeah.
Thank you, Mohan. Any other question? Again, those young kids are, oh, very good. Wait a minute. Okay, she got the mic. So don't you listen to me, see me, and I ask you whom to give the mic, okay? Please. That's how it should be done. Okay.
The last question, sorry for the interrupt. Actually, my question is to Gopi sir. So, we say prevention is better than cure. So, why can't we eradicate the cancer with the help of molecular biology at the same time curing it? So, like we use polio, like vaccine to cure, eradicate the polio. So, like, why can't we do with the help of molecular science to eradicate the cancer
Yeah.
instead of curing?
Sure. That's what I was telling. So it's one is people are looking at the vaccine for certain cancers. Not all the cancers are not possible. But see, that's what I explained in the beginning. We are made up of 38 trillion cells. We are all exposed by all kind of chemicals, pesticides, whatever. When you go out, some cells are dying, some cells are getting mutated. All things are happening around us. So some cells transform into something, a different form. That's how the cancer is. In fact, I was talking to a doctor in the CMI, Aster CMI. He says after 80 years, if you do the biopsy or autopsy, if someone dies, like there will be 80% of the people will have certain tumor or some kind of a cancer growth, it seems. So that indicates this is there, but people are not able to identify. It is because it's a mass of cells which they want to grow or which they want to form a kind of a community indirectly. So but that is not harmful many of the times. It's coming and then going, and the mutations are happening and going. So you're not able to find out. But sometimes if it happens early, that's where you have the issue. So that's a lifestyle disease rather than the once you are, now that we are all living for longer time, that becomes more evident, because earlier people used to die a little bit early. They never used to see that. So now that we have a lot of medical awareness and other things, that's another bit.
Very good. And I appreciate that. There's one young, question by this young lady here.
My question is to Sailaja ma'am.
You please introduce yourself better.
My name is Kunchita. I'm doing a PhD in biotechnology in GKK. So my question is it possible to cultivate this seaweed in mainland? So like, can we mimic the physiological conditions of this seaweed and cultivate it either individually, separately, or can we integrate it with other aqua farmings like shrimp cultivation or pearl cultivation? Can we integrate with that?
So actually that defeats the purpose, or why we are going away from land, because then you will be taking land for doing seaweed farming. Also, there is a huge issue with shrimp farming, the way it is done. You're actually using mainland agricultural land. You're putting in salt water to grow shrimp. This is how it is done in all of Andhra Pradesh, and it makes the soil saline. So you cannot plan on doing, I mean, for me, it is illogical. I mean, I wouldn't think of that.
No, because you said there are so many government regulations and hence the production is very low. So
Sorry,
can we? You are telling us there are so many government regulations, so that production is very low. So if we can integrate with the
production is low because we don't have shallow waters where seaweed can be grown, and nobody has even thought of technologies to grow things on the ocean. So if we have the ability to demonstrate that, we can demonstrate even in India with our technology. But there are other core issues in Indian legislation because of which, even if we grow large amounts, we can't export. I mean, there are some fundamental issues in Indian legislation which I cannot discuss. I mean, right now is not the forum. Yeah.
So how long, like in how many days do you harvest this seaweed?
In 45 days, it grows 4x in 45 days.
Thank you.
Seaweed farming can be very remunerative. Think about it. So,
best bank.
Yeah. It's okay, Oman.
I would ask.
It's working.
Okay. So I would ask to Dr. Gopal Krishnan, because he is having the background of both plant and animal. So since I am not directly from physiology, I have always have a curiosity that, see, if we go to a doctor, they will suggest some blood test and pressure and all those things. So based on that, they will do some its diagnosis. So for plants, say for example, high value plants like kesar, vanilla, or maybe plants having high gestation period, can this be done? Is it possible? No, not blood pressure, some kind of that kind of test.
I think that's a good question. But yeah, I don't know the fluid. If you look at it, the plants also exclude some fluids, isn't it? It could be just if you scratch the leaves, you might get some water coming from that. I have not checked. We have not.
Yeah, I mean, such diagnosis that we do very, very simply is to measure the turgor, measure the surface temperature, finished. So if the turgor is good, things are okay.
Yeah, so maybe nutrients and then some of these things. Yeah.
Yeah, nowadays, but then they do a MRI scan on the root to find out how big the small the roots are. There are a lot of things that are being done. Use the medical science in the plant science, which comes much later, which is happening now anyway.
Just to add to that, people are trying to develop these microneedle patches, but then collect the interstitial fluids.
Yeah,
To
that
use
is
them
what
as a diagnostic,
uh,
but pretty early stage. I haven't seen much work, but that's one approach
because
people are
my protein is also located in the
Especially for
apoplastic space. So
Especially
I
for
would be very
infections.
interested in doing that kind of test. Okay, thank you.
Bindu Madhav, you have a question? Switch it on.
Yeah.
Hello, this is Gopal Krishna. It's always good to interact with you and I come to your company. It's exciting to visit. So you are from the plant background, and after that you joined with a couple of companies, Nitrogen and also Genotypic. When you started your startup, your plant science knowledge or background helped you, or the couple of companies where you worked that helped you? Because from the plant science background, if you go and talk to a medical science, researchers or doctors, particularly in the cancer, so probably how the response and how the interaction, because they know you're from the background to plant science, and also distinguish that also, and how probably initially you felt that that interaction, how actually the responses from other sciences.
I think, yeah, that's a good question. So we always have in the back of our mind, like where we come from. A lot of the times, sometimes that inhibits what you want to do. So sometimes I feel experience is sometimes it is more inhibitory rather than the exploratory approach what you take. So what in fact, when I moved from scientist to sales side, what I did was I removed my doctor thing there. So in all my business card, I used to just put my name, saying that people, even if they're talking to me, they should think I'm a dumb person rather than like this guy does he know or anything kind of thing. That's the route I used to take. And another thing is, once you talk to the people, and then once they understand what is your depth of knowledge, they don't really, in fact, they don't really bother about those things. So the initial interaction might be a little bit difficult. But obviously I have interacted with a lot of doctors and other people. So they take it open. They don't even ask us where. They look at our company and then what we are doing. They talk about that. That's what I see. So branding, it takes some time. It could be your personal branding or your company branding. You have to project yourself what you want to do rather than what you are coming from. That's what past you should just leave and then just. But past is it helps at certain level. As a business person, you have to use wherever you want what you want. So that's how I look at it. If I'm using if I'm going to agriculture university, I have to use my plant background that we have to use. And then if I'm going and talking to doctors and other things, obviously I connect where I have worked with the doctors. So how you use it matters a lot.
That's a good question, Bindu, because there is one very nice statement by some author. I'm not getting the name of that, but he said, if you know that you are an idiot, it is better to keep quiet and let them think whatever it is than to open your mouth and prove them right. So it's a good thing that he has not put the doctor, which doctor, plant or human doctor. So that's a good idea. Thank you. I think there will be a lot of questions. I think we are running out of time. I think we should wrap that up. So is there any final comments? Is that look, this system is very interesting in the sense. I mean, one thing that I'm really happy about is that there are so many questions asked, and I'm really happy about the questions that you asked. Questions, that is all. Whether the question is good or bad or ugly or beautiful, that doesn't exist. So ask, and there will be some good answers. And therefore, I think our panelists have given us ample information today of how hard it is, or how nice it is, or how easy it is, and how beautiful it is at the end of the day if you succeed in a startup ecosystem, and what needs to be asked. If you are asking for the funding from venture capitals, adventure capital, you should be adventurous, asking a very, very strong question and tell them, this is the reason why I strongly believe this is going to work. And once you have that database, I'm sure things can be very easy. And
I think when we started in 2010, this entire ecosystem of grants, initial grants by government was not even there.
Correct.
Okay. We actually had a very hard time in the first 2-3 years to even secure funds. We had to reach out to alumni of IIT. We were trying to do like high net worth individuals, and we we're trying to raise angel funding and all of that. I think when this fund schemes like what big is coming up, at least till you get POC or proof of concept, you are at least, you have somebody who is holding you or who is like helping you out. I think these kind of schemes and the kind of mentorship and ecosystem, at least in CCAMP or in US, I mean, you are also well connected with the entire ecosystem. I think this kind of fosters people to at least think and say, okay, can I try this idea? Even if I fail in 2 years, I can go for a PhD, or I can do something else. I can get a job. So I think that leap of faith sometimes is only possible at this stage, I feel, when you're a student. So if there is a appetite, people should definitely explore entrepreneurship with concepts and with all the due diligence before getting in, because it's not an easy ride. But yeah.
Fantastic. Because in this big.
I'll add one more point. So again, see, a lot of the times when you're young, you can try a lot of things. Okay, as you grow old, the risk appetite is very low. So because you already have certain comfort level you have taken, and then you are not able to break that easily. So if X or Y getting high salary, they will never go to the trying the thing. So another thing is, Shailaja said about the grant and other things. In fact, I never tried. I tried once or twice certain things, but a lot of the times what happens is I would try to validate in the customer place rather than sometimes grant side. So just go to the customer and validate. It might be easy because sometimes grant is also a vicious circle. You kept on getting behind the grant. If somebody doesn't give the grant, don't lose your heart. Just find the right people, and then it could be a smaller scale. Even 1 lakh rupees you can find from some people. Some 10 people can join and then start creating the prototype. Once you have the prototype, people can fund you easily. So this is another thing which you guys have to think of it in case if you have some funding issues. So that's.
Thank you, Gopi. I think Praveen, you want to say something at the end?
I think like there are a couple of points I echo with Gopi. So in fact, I'm not very big fan of grants. The reason is, one is, but they are non-dilutive funds, right? That's the best way to raise the money if you wanted to. But the question is the validation, right? Like, me convincing Bayrak is a lot more easy than convincing Aman for write a check, because that's also gives you the validation of the technology. Does it have? Because as a science, I can put like a very creative idea and convince other people, but like, does it have relevance? Does it have that practical application, and it has a potential as a business case? If I don't see that I have to do it much earlier than much later. So that's why like your raising project money, like a dilutive money, you might lose some fraction of the company, but like that's the best validation you can get it. So one need to see like what you want to gauge and like, how you want to balance it. Yeah.
That sums up the whole thing. And it is important that we get to convince the capitalists. And I think that's a bigger challenge, as you very rightly said. I was talking to Rakesh Mishra and other guys in CCMB. There are only three people you need to accept your concept. One editor and the two reviewers. If these three people agree to you, your concept is printed. Whatever happens, that doesn't give you money. So these guys will ask 1 million questions. And when you answer those, and I'm sure there is no end to the sky. So with that, I think we should close this. But before we do that, I would request my colleagues, Dr. Mohan Raju, Dr. Nataraj, and Bindu Madhava, and Prakash Patil, each of you please come and present the plaque to one of the panelists, please. You can choose to whomever you want to give. I knew that. You have to give me money for that. Ilbani.
In fact, thank you.
This picture will come in all social media now. Gopi and Bindu. Yeah. Why don't we all stand here for a picture? Photo. Bye. All the press people have taken enough pictures. Thank you. So, Akshata, will you tell anything? Or so, as a last part of this concept this year, may I request my friend Dr. Mohan Rammohan to wrap up today in 10 minutes.
Good evening. Yeah, before I forget, let me first thank all the online participants. If you're still there, thank you very much. It has been a great one day, great concept 2024. Let's first start and give a big shout out to Shesha and team for putting this together. I think September is a very important month. I think we started the month, September 5th happens to be Teachers' Day. We think about gurus. And today we are celebrating two legends, gurus here. And I think, as Shesha mentioned in the morning, very rightly so, while we are talking about Professor Shastri and Uday Kumar for their contribution, we also need to acknowledge the contributions made by Ramarao sir, and I think Shesha did that very well. I won't go to the, though I have, Shesha, for your whole sheet of notes here, but I won't go to the details of any of it, just because the presentations were just great. Professor Raghavendra, one of the close friend of Professor Uday Kumar, and somebody who regarded Professor Shastri, set the tone on talking about photorespiration. A key message, photorespiration is not an evil, it is a necessary. And second thing he summarized is that it is better to look for strategies which are more on working on the plant architecture type rather than really tinkering on photorespiration, right? There are two key messages. Then, of course, Professor Chinnuswamy Vishwanathan, I wanted to tell him actually, but he left. His work on stress tolerance, especially in Professor Zhu's lab, early 2000s, I think every other Thursday meetings in university or in the Department of Physiology was understanding their papers. I used to come from Monsanto after my work. On Thursdays, they would professor would be talking about, or a group of students. I still remember Sendil, perhaps Rame around there, talking about Zhu's papers and Vishwanathan's paper. And Professor Vishwanathan, please note, our students have been stressed by your stress tolerance papers, okay? But please. And of course, he did a great job presenting about reconstructing the whole ABA biosynthetic pathway, the complexity involved in it. But at the end, he kind of connected it so well with the topic of the day, the biostimulants. I think that was a great aspect of it. And of course, after that, Soham Ray, you spent some time about talking about your work. Yes, it is not on physiology, but it's something which is probably going to head way towards the next future, especially genome editing. And I like that idea of working with not one, but a few of them at one time. And I'm sure Professor Uday Kumar would have been more willing to kind of listen to you more about it. Then we had the break. That first session was the cake, and then the next session was icing on the cake. It was just a blend of having all the entrepreneurs coming here, sharing your experiences with us, talking through your success and challenges. It was excellent. And the cake was, the cherry on the cake icing was those two students who came and presented their proposal. So all in all, at least I believe that this is perhaps the best editions of Concept 2024 concept so far. And mark your date, September 27, 2025 will be the next one. And I'm sure Shesha and team will be back to the drawing board, scratching their brains, what should be the theme. So till then, all the very best. See you next edition. Take care. Bye-bye.
So before we wind up, somebody who's online want to say something about the Concept 2024, please.
Maybe can you hear me?
Sajeevan, we cannot hear you.
Oh, now? Can you hear me now? Is it possible to hear now?
Yes, go ahead.
Yeah. I'm Sajeevan. Currently, I'm in Swedish Agriculture University in Sweden now. So it was quite happy to see all the old faces in one platform. And in fact, I got the opportunity to work with Professor Uday Kumar for some time. And then And also got the opportunity to work with Dr. Shailaja's project when I was working with Professor Nataraj Karaba when we are having their first product testing. In fact, I was involved in that. And joining with NCBS, Praveen was working next lab, Professor Saudamani's lab. So I was hearing all the exciting stories there. So coming to the concept, it was one of the best ideas, even though Professor is not with us, bringing young brains with connecting people, what all different opportunities as a student. In fact, agriculture students always have a thing that they don't have much opportunities. So you can see where all you can move forward after your studies or during your studies with the connection with all these ideas and in your academic field. So I wish you all the very best for the concept, and once more thanking all the people who has taken this initiative for bringing this and connecting all the people together. Still, Professor is not there, which one of his biggest dreams, which we all are carrying forward. Thank you.
Thank you, Saji. I think if you can, Nirja auntie, you're there? Oh yeah, they are there. Uday Kumar there. Soumya, Soumya and Nirja auntie, I think they are there.
Okay. Any minutes.
Huh?
We were waiting.
Yeah. So when we are waiting for Nirja auntie to talk, and is there anyone else who wants to say something from the online audience, please?
Hello.
Auntie.
Sesha.
Yes.
I had actually sent you a message a few minutes ago.
Oh.
But anyway, the gist of it is that I'm very, very, I'm very gratified that, this concept edition also has gone through wonderfully. And I have sat through the whole session right up till now. And though many of the concepts were beyond me, I identified with the spirit in which it was conducted, and congratulate all the participants, including your young students. So all the very best for the next concept 25.
Thank you, auntie. Would you mind opening the video for a minute at least? I
have to check that out, Aditya. They want the video.
Talking to
Video.
Mrs. whom we call Nirja auntie, and I don't want to say more. I'm emotional. There you are. Oh, Avanika did that already. Wow.
Yeah, I say hi. Come.
Hello.
Yeah, she was here for part of the session.
Yeah.
Okay, and Soumya had to leave, though she was also there for much of the program today. So all the very best, and please convey my congratulations and thanks to all those who are responsible for this great success. Thank you very much.
Thank you so much, auntie. I think that is the grand finale of this. Getting a blessing from Mrs. Professor Uday Kumar is something which is extremely beautiful for us. Thank you, auntie. Thank you so much. I think we will close now. And thank you all very, very much. And all the people who helped me, I should definitely thank the entire student fraternity of the Department of Crop Physiology. Thank you very, very much. I don't want to name anybody. Anybody is talking there online?
Hello. Hello.
Oh.
Hello.
Okay.
Yeah, hello. Yes, we can hear you, sir. Please.
Yeah, this is Dr. Sandeep Pawar from Nasik, Maharashtra.
Hello, Sandeep. Hi.
Sir, it was a wonderful event. Actually, I'm a biochemist mainly, and I've worked on cancer biology and now serving in the agriculture industry. So I can correlate with Dr. Gopi sir and the event. It was a wonderful event and thought-provoking event. And it's a US organized event. So I have always have that this thing. If there is any event of US, I have attended it. So it was a wonderful event. Thank you so much for organizing such a wonderful event. Thank you so much.
Thank you. Thank you so very much. All right. So thank you very much. As Ramon said, see you on the last Saturday of 2025, September, again.
Yeah, we have coffee, tea, and light snacks served outside. Please have, and please do join us for dinner at.
But just a second before we finish that, Rame, you please come down and give the plaque to Dr. Ramon for wrapping that up. So I'm signing off. What did I say? I rest my case.
Please do join us for dinner at Chairman's Club, 7 o'clock. Time, sir, yalla, sir? 7 o'clock, time. 7 o'clock dinner, please. Sir, please do come.
Zoom end